by Professor H.A. Ranganath.
No, Upendra, just for fun, I said. Please go ahead. Just for fun. Good luck. All the best.
Okay. And for today's sessions, we have the two session chairs, Dr. Amritavalli and Dr. Shakuntala. Dr. Amritavalli is the associate professor at the Department of Studies in Genetics and Genomics, and she specializes in bioinformatics. Dr. Shakuntala V is associate professor at Department of Studies in Zoology, and her area of specialization include chronobiology and Drosophila population genetics. With this, I welcome you all to this today's sessions, and also I welcome the session chairs to continue with the third session of this conference.
Good morning, everyone. I have a small request. Those who are occupying the last seats, please come forward. Please occupy the front rows. I would say it's a very happy occasion, and indeed I am privileged to introduce Dr. Upendra. He is a professor in the Indian Institute of Science, Bangalore, and he got his PhD from University of Mysore, and he has made seminal contribution in the field of muscles and neurobiology by using genetically traceable model organisms, fruit fly Drosophila, and zebrafish Danio radio. He has been serving as associate editor of the Journal of Genetics. He is life member of the Indian Society of Cell Biology, Indian Developmental Biologist Society, Society of Biological Chemists of India. He is recipient of Sir C.V. Raman Young Scientist State Award from the Government of Karnataka. Over that, I would like to add one or two sentences that a teacher specifically will be satisfied, or it will be a great achievement for the teacher when his student is going to achieve great heights. Similarly, the legacy of students attaining great heights is seen in the Department of Genetics and Department of Zoology. Professor H.A. Ranganath's student, N.B. Ramachandra, the legacy is continued, and Professor N.B. Ramachandra's student is Upendra. So I would like to say that he is a proud grand student or grandson of the department, right? So I request, I have a message for you students. You people should join IISc and you should continue the legacy. I want minimum 5 of you to join, come say, clear the gate and then join IISc, join under Upendra, and then one should continue the chain and one should continue the legacy. See, the greatest satisfaction a teacher is going to have is seeing the success of the student. And this is how Professor N.B. Ramachandra's achievements and Professor Upendra's achievement is well received in the department. So with these words, I hand over the session to Professor Upendra to start.
Good morning to all of you, offline and online. Thank you very much, ma'am, for your kind words. Without wasting too much time, I will start my presentation because we should finish in next 20-25 minutes. I have prepared the slides for maybe 45 minutes, which I was given, but now I will finish in 25 minutes, right? But before I start, this is for the students, right? In China, there is a city called as Guangzhou, and if you go to that place, the sun rises at around 7:30 in remote place, right? Slightly away from the city, there is a Guangzhou State University, Technical University. If you look at the timing, 7:30 is the sunrise time, right? Unlike here. the students are in the lab by 8:30. Whether they have function, no function, or anything, they are at lab by 8:30. All right, all of them. Most of them, almost like 95% of them, they can't speak English or write English. But you look at the most of those students, they will have a review paper by the time they complete their masters or the first 2 years of the PhD. Because with tuta futa, we call it, right, sir, in Hindi, I mean, they will write the review, then their PI will correct it. Even though those corrections are very bad, by the way. Those who are, I mean, I have an associate editor with genetics, and we get almost 95% of the paper from the Chinese labs, actually. So most of the English are really, really bad. Okay, what we call bad, our bad is good for them, actually, right? But they are so committed. Today, till 1985, India was much ahead of them in terms of the publications, the science and everything. Today, they publish almost 3 times the Indian science. Not only, some of us, we will say that they are Chinese, that they are mass production thing, right? Okay, but it is not true. They publish in the quality journal also. You take any, say today, Nature, Science or Cells or any journals, there will be Chinese papers, okay? They have invested extensively. But it is not only about the money. You see, if we invest too much money on you also, you will get spoiled. Right? Yeah? I can tell you, you just have a look at the most of the professors' sons and daughters, right? We work hard to become something where we are. But we have given them so much privilege that they would have struggled to become something in life. Is it true, Subramanian sir? Yeah. So, when you are privileged, you don't become something, right? Okay. And I have been asking you yesterday questions, right? The teaching, I mean, in India, we teach you everything and you expect everything from the teacher. So you stop thinking. Right? And so today's question is the simple thing. Right? Yesterday I asked you 2 questions. I'm going to ask you simple questions. Again, in the central dogma, let's say DNA to RNA is transcription, RNA to protein is the translation, right? Let's say that we don't have the translational, that step, the RNA step. Now, you can have the protein from the DNA directly, right? You tell me afterward, what will be the advantage of having such a system and what will be the disadvantage of having such a system, right? Yeah, is that okay? See, it's just a game, just playing around, just thinking, I mean, right? Okay, we'll do that. And so I start my talk. Today I'm going to talk about, I like walking, so I will walk around, right? Okay. So we'll talk next. So I'm going to talk about mitochondria. Yeah. From the day we knew, right, biology, right? In biology class, you have been learning about mitochondria, right? And when I became PI, I thought that we know everything about mitochondria, right? We don't have to work about it, right? Yeah. Today, one of the most exciting field in biology is actually mitochondria, right? And I'm going to just share our excitement with you. Few things, right? Then you think about it that you take still any organelle, cell organelles, you can learn a lot. You have tools and technique now to relook at what we knew about these organelles, what is their contributions. So we are just refining the science. Okay? Maybe on the way we may do some breakthrough research, but over the years now we know about mitochondria. Why we got excited, I'm going to share it. So you don't have to look at the data. Data are generated, you can generate. But just try to understand the essence of the science, why we have done it, right? Okay, so you all know that mitochondria is the powerhouse, the engine of your car, right? Yeah. So depending on the type of the vehicle, you have 350cc for the bikes. Yeah? If you want to carry big ones, you have trucks, so that you need big engines, right? Likewise, mitochondria is also of different size depending on the which organ. Next. So today we know these are the major things. Today, if you look at any cellular activity, mitochondria is required. Of course, we know that it is because it provides energy. Beside the energy, today we know that actually mitochondria play major role in how they move, how they survive, and things like that. Classical example, if you know it, Alzheimer's or Parkinson, if neurodegeneration, if you look at, the mitochondria actually get stuck in the first place because they need to be transported from the cell body in the any axon, right, to the end, right? Yeah? Okay? So if anything transport hampers there, that is where your neurodegeneration and other things happen. Okay. Similarly, you look at anything, what we knew is that it is required for the ATP synthesis. But today we know that in the oogenesis, spermatogenesis, your stem cell pluripotent maintenance of the pluripotent, everywhere mitochondria is required. Okay. It means that they are playing much more than the energy, just energy product. Okay, so we'll look at. And the exciting thing is that we have hardly 16,589 base pair DNA, right, for the genome of the mitochondria. But if you look at this C element, it has got almost like 8,000, 81,000. Now you can figure out why they need to have this, right? Okay, but because the mitochondria was, like mitochondria genome, I mean, per se, that it takes half of its functional of the individual genome, right? Individual genome means the nuclear coded genomes, right? So, as I said, you will find different kind of mitochondria. You will have the lung tissue, usually will have this roundish mitochondria. Liver one will have the elongated one, right? And the kidney, you can see the little disc crystal like mitochondria, right? This tubular retina of our cone, right? They will have the reticulate mitochondria, right? Yeah. So there are different sizes and things, right? And so in the resting conditions, you can see the mitochondria crystal, they again will be reticulate. So they don't form oxygen and things, and don't sleep yet. Yeah? All right. Yeah? So you see here the difference. This crystal becomes alive here when they are active. Okay? And this is the whole point. This will be elongated, nice cell survival. If they are big, then they will be elongated. That will enhance the cell survival. The moment they become fragmented, they will be small and they die, right? And this is the whole point. Dynamic in the sense that mitochondria, whatever happens, it will be elongated, then they undergo fusions, they will be big. Then depending on whether if you are using your muscles or not, or neurons or not, if you are bedridden, your mitochondria will become fragmented, right? Okay, again they can fuse. As we get older, you will get more fragmented mitochondria. And today we know that if you have read science paper a few days back, they have published that if you remove, there is a protein called the PGLA, and if you remove mitochondria, right, activity increases, and the mitochondria, fragmented mitochondria, if they are removed from the cells, right, the individuals survive much longer. It's okay, right? Today we know that mitophagy is very important thing. Mitophagy, during the early development, if you have the mitophagy, it is not good, right? Removal of the mitochondria, okay? It's not good during the early development. But in the later stages of your life, like those who are diabetic like me, or those who are old, mitophagy is good. The unused mitochondria which have, done its function, they should be removed from the cells so that the individual can survive, right? So that you can have increased longevity or the health span if you have say, these fragmented mitochondria are removed, right? Okay. And I will come back to you this afterwards. We have identified the molecules which make it fragmented, okay? And you can play around with these things. We are looking at the moment pharmaceutical interventions which can make it, keep it in the elongated condition, not in the fragmentation condition. And we also identify, I'm not going to talk about it, we also identify a phosphatase. Ramachandra sir, student Hirojit, identified that phosphatase. Now today we know that it is involved in the removal of this mitochondria, non-functional mitochondria. But that paper we could not publish. Before we publish it, somebody published it just recently, okay? Right? That is where the competition is. Science is highly competitive. You cannot just say that, we have the data, nobody will overtake us now. Somebody will always do it. Right? Similar kind of work, similar kind of thinking. So if you are late back, somebody will publish before you. Right? We have been having this data from last 5-6 years. We haven't published it. We thought that nobody can get the data like us, but 2 weeks back, the paper came out from a Chinese group, right? Yeah, they have nicely referred our first report. And that's it. So we can't publish it today. I mean, we have to look at the pharmaceutical thing, then we will publish again. But you need to have the novelty, right? That novelty is lost. Somebody overtook us, right? Okay? Yeah? So while you are sleeping, somebody is working very hard. I mean, think about it. Yeah? All right. Okay. So mitochondrial dynamic is like, when completely mitochondria is done with the function, as I told you, this will fuse, this is the one you want it, but once it gets older, it needs to be fragmented, and once it is fragmented, it is done with it. It needs to be removed, and this is what we call as the mitochondria. So what controls all these things were not known, and you can see that what we know from the thing is that there are 2-3 only proteins, and their role in the mitochondrial fusion or fission is very well known. So you can see the mitofusin. These are the one which are the outer membrane protein, and we have only one. This is in the vertebrates, but in Drosophila we have only one called as the MAR, and the inner membrane ones for the fusion is opal-like. Opal-1-like is the inner this fusion for the membrane fusion. Okay, these are the mitochondrial protein required for the fusion of the mitochondria, right? Then you have the DRP1, dynamine related. These all are the small GTPase proteins, okay, right? So you need DRP1 for the fusion. Once fusion is over after functioning and everything, DRP1 will come and you will have fission, right? So you have this thing in Drosophila, it is again ortholog is the DRP1, and people have worked extensively about how it is involved in things. For us, however, the important thing is that what regulates this, that you need to now fuse and you need to now divide, right? Because depending on the cell's function and the thing, it has to be highly, tightly regulated. Is that okay, right? And the role of these proteins are well known, but what is not known is that a tissue-specific way how they are regulated. Because this identifying this regulating protein is important if you want to have an interventional thing. Like diabetic patients, right? What we call as the remodeling of the mitochondria. Today, diabetic people or people with the Alzheimer or the things, they have completely different mitochondria, right? So if you want to intervene, other than we will look at the particularly organism level phenotype, that is the aggregate. Much before, if you knew that these are fused or fragmented, how we can intervene here, that is what we are looking at, the molecular player, right? Which will regulate the fusion and the fission, right? Okay, that is what we wanted to look at, and muscle is a good thing to look at, because if you look at the muscle, depending on the tissue type, right, slow muscles or skeletal muscle or the cardiac muscle, you will have completely different mitochondria in the first place. And depending on the risk, whether you are suffering from something, the mitochondria will change in the shape and the size and the thing. That is the dynamicity of the mitochondria. Okay, so let's not talk about it too much, because we know today that every disease condition, mitochondria is the first one which get affected. Okay, so it is very, very sensitive. Unlike other progressive phenotype, what like say brain atrophy and things, which you can get it after say 5-6 years, even after the Alzheimer's starts. Unlike that, mitochondria actually you can get on the first day itself. Is that okay, right? So this is very important. So we work extensively in the Drosophila flight muscles, because some of the mitochondria is 3.2 micrometers. It means as long as the sarcomeres. Okay, the flight sarcomeres are the one of the biggest sarcomere, right? Longer than the human, and some of the mitochondria you can see it. It's longer than actually the muscle sarcomere. You can see it. Yeah. Sarcomere is one sarcomere, some of the mitochondria. And today we have technology like say cryo-EM and the things, sectioning, where you can do beautiful imaging, unlike before. Today you can really count each mitochondria actually, right? Yeah, which was not possible, right? People just say that 10 to the power 4. Anything between say in the yeast, 10 mitochondria per cell to, right, you can get up to 10 to the power 4 in liver and things like that. Okay? But today actually we can count, right? And we can just look at the tissue specific thing, how they are regulated. So my student who worked extensively on this thing in my lab, he is doing postdoc, and he has identified a regulator of this muscle, the flight muscle versus the leg muscle, for example. Okay? So still you can have, in other words, I mean, you can still have a career out of something which you have been studying in our time from class 3, right? In your time, maybe what time, I mean, I don't know when they introduced mitochondria, right? Yeah, but I think it's the 5th standard now, right? Okay, but people can still make a career out of it, right? Okay, that's the take-home message. So we work extensively on these flight muscles because it has got the large mitochondria, as I told you, and it can be, my time is over. And these are papers published already, whatever I'm going to talk, so you people can just go through it. So why we work extensively is that you can just look at the spatiotemporal during the development. I mean, how they fuse, what is the mitochondrial shape, you can see that, and then how they grow, increase in the size, everything you can follow it, okay? So that is what we have done extensively by using confocal microscopy, then electron microscopy, and now today we have the topography and the things which I haven't included here, but I show you the example there, okay? So you can study all these things, and first thing is that we wanted just to look at how these things happens when you remove those 3 proteins, right? Fusion and the fission. Fusion is the one, MAP1 and the OPA1 like, right? Because if you want to look at how it is regulated, first thing is that you look at the phenotype, right? Yeah? Is that okay? My approach, is that correct, you think? Yeah? So we did that. We knocked down this protein. You can see that. First, we look at the expression. Expression as we go during the myofibril. This is the adult stage. So it is highly expressed. Just remember this timing, around 70 hours after the puparium formation in the fly. Okay, so that time it goes up. So what you have to do, what we call as the myofibril, that is where the protein assembly of the muscles happen. So this relates with the myofibrillogenesis, right? So what we did was that where it is highly expressed, you knock down at this stage, then see if there is any phenotype, right? Where it is highly expressed, the obvious thing is that you knock down at that stage and then look at the phenotype. And that is what actually we did. We knocked down. You can see the knockdown here. Let's not look at the quantification, okay? So, and we just look at the phenotype. phenotypes were almost comparable to the wild type. Even the protein is expressed, protein is highly now knocked down, it doesn't show any phenotype. Right? Flies are flying very well, no problem at all. I mean, slightly things, everything is fine. The same thing we did it with the Opa wildlife. At the same stage, we knocked down where they are highly expressed, and again, we didn't find any difference. Okay? So if you knock down where it is highly expressed, there is no phenotype. Amazing, isn't it? You expect that the protein is highly expressed, you knock down at that stage, you should see phenotype, but we didn't see it. Now what we did was that then we look at the earlier where it is, they are not highly expressed, but it is expressed, they are expressed, but not very highly. So we knock it down at that stage, you can see the knockdown, and you can see that the mitochondria, once they are supposed to fuse to give rise to this, they remain fragmented. In the earlier one where they are, expressed very low, at that stage, in the low expression stage, if you knock it down, you see fragmented mitochondria, which you can see it here, okay? Highly fragmented. So we call it as a circularity. If you look at the mitochondria, how circular they are and things, you can find that the circularity is lost in all these flies, right? Okay, let's not depend too much on things. And we know today that if they remain fragmented for the long time, they will undergo mitophagy. In other words, they will be removed from the cells, right? Okay, and that is actually what we saw: increased mitophagy in the muscles. Okay, you can see it here. Yeah, they will not fly. In the early one, they don't fly. Because the muscles are abnormal, mitochondrials are abnormal, autophagy, everything, so they don't fly. Yeah, you can see it. 100% they don't fly. Okay. Yeah, so today we know that even the sarcomere, even if they survive, they are very thin. And today we know why, because the protein synthesis again is affected here if you have a smaller mitochondria. And again, this I'm telling you the thing. We had the data. My student left my lab to do postdoc, and we haven't published the mass spec. And another group now they have published that data. Okay, yeah, so it happens, right? Okay, yeah. So one group from France, Snorer group, we are competing with them, but they analyze the data and they have published it in better journal. Right? Better means in terms of impact factor, but we are the first before them to publish it, right? So fly, less fly, the same thing we got it with the OPA one, like, right? Inner membrane one, you knock it down, you get a mitophagy in the developmental stage, right? Okay, this is the take-home thing. Mitophagy, you can see it here. Okay, so these two proteins, inner and the outer membrane protein, if you knock down when they are highly expressed, you don't see any phenotype. You see the phenotype only during early developmental stage where they are slightly expressed, right? So the question is, if you knock down in this stage where they are highly expressed, you don't see the phenotype. You are knocking down in this stage where they are just coming up, then you see the phenotype. Now the question is that what regulates this stage? Their maintenance, that small thing. That is what our major objective is. Okay. So the other one is the DRP1, the fission one, because there is no fission of the mitochondria. Early one there is, but there is no much fission in the thing. So when we knock down the DRP1, there is no phenotype. So DRP1 doesn't give phenotype during the development if you knock it down. Okay, that is the take-home message. So now what we are looking at is that this is the final thing in the DRP, MRP and OPA1 like, you may have a small mitochondria and they undergo mitophagy, and in the DRP1 there is no function. Okay, so the question is that what regulates this fission and the fusion during the development? That is more exciting because that is the one which can be targeted for the therapeutic use, okay? Not because OPA1 like if you target or the thing, it is all over the body. It is not tissue specific, right? So if you want to do a drug therapeutic or something, you need to have tissue-specific things, right? And it becomes easy. And this is where we discover the erect wing. Erect wing is a autolog of nuclear respiratory factor in vertebrae, okay? And we saw that the people have, including Professor Vijay Raghavan, they have published that the erect wing actually give rise to this wing up phenotype, okay? And they give the muscle difference, and that is what they have shown. But what we know from the vertebrate study is that the NRF is required for the mitochondrial dynamicity, right? So erect wing is expressed in that early stage, not after the muscle differentiation, not after the muscle that I was talking to you when the upper one leg and things were highly expressed, right? So erect wing is expressed only in the early stages. After 37 hours, actually, there is no expression of erect wing. Okay, so this is the erect wing expression. And so when you don't have the erect wing in the muscle, what happens is that, again, you see the phenotype just like you saw in the knockdown of the those upper one leg and the mark one, right? The mitochondria remain small and they show mitophagy. Okay. So this is similar to the one where you saw the mitochondrial phenotype. Okay, so that is where we look at its function. Mitochondrial JC1, we call it, membrane potential is less and everything. So mitochondrials are not functioning properly, right? So let's not worry about all this. So what we are looking at is that we look again what happened to all these proteins which are required for the mitochondrial fusion and fission. That is where we found that actually erect wing regulates the upper one leg. You can see here, upper one leg is not expressed, right? If you knock down erect wing. Whereas you see MARC and the DRP1, they are fine. Okay, so it means that upper one leg is regulated by the erect wing. Now you think about it. Upper one was required at the later stage. Erect wing is in the early stage. And when you knock down the upper one leg in the early stage, you see the phenotype, right? Yeah. So that shows the whole thing, right? Upper one leg, you knock down only in the early stage that you see the phenotype where erect wing is expressed in that window, right? So what we are doing is that the erect wing is expressed in this window. If you upper one leg here, it is not expressed. Upper one leg is expressed very strongly here, right? So if you knock down upper one leg in this stage, the flies are normal. But upper one leg, if you knock down into this stage, that is where the degeneration happens. So it means that the regulation, the activation of the upper one leg in this stage is important by the erect wing for its function here later on. Are you clear? We presume that the protein should be expressed at the time where they are supposed to work. But what I'm trying to propose, right? This is our work and this is our contribution, is that you need not do that. You can be regulated in the one window, which is almost like, say, 40 hours before, right? And that protein will be required after 40 hours. So the protein upper one leg need to be activated here, that's all. But that protein activation is required in this stage. So if somebody tells you that proteins are activated only when they are required, it is not correct. So this is one example. Okay, where you can have a protein activated in a slight way in the different window for its function in the later stage, right? Okay, that is what our whole contribution is. You need upper one to be activated this stage so that it can have normal function in the later stage after 40 hours, right? And so what happens if you have the mitochondria small, right? Take home message is that here there is a condition, right? You see the mitochondria, we keep the mitochondria very small. As I told you, if you keep the mitochondria small, the tissue will undergo apoptosis, and that you can see that muscle is completely undergoing apoptosis, right? So mitochondria is very important even when it is not functioning. We have done lots of experiments like cutting the wings and keeping it so the mitochondria, they don't fuse longer and things, and what happened? And Drosophila is slightly different because if we cut the wings and keep it, just like that, actually they survive better than those which are flying around. Yeah, and that is because whatever said and done, there is not much regeneration once the muscles are formed in the insect or mitochondria, in the Drosophila, compared to the human, where our muscles, proteins are changed every 3 days. The thin filaments are remodeled every 3 days, and the thick filament, myosin ones, which Sandy talked yesterday, thick filaments are remodeled every 10 days. So if you are bedridden for 10 days, you will have completely different isoforms, right? Which to survive, to sustain your energy, then compared to the one who are running today. If you start running today, you will, within 10 days, you will have a completely different isoform. Right? Yeah? Okay? This is the take-home message. So what we can learn, there are slight differences between Drosophila and the things, but you can learn a lot from the Drosophila. And what I have tried to convince you today is that there is no harm in thinking in a different way, and still you can contribute new things. Yeah? Right? Yeah? Okay, thank you very much. I stop here. And these two work was done by Mamta first, and then Mamta was supported by Kati. And Mamta has been working extensively in the St. Jude's Children's Hospital, postdoc, and her contribution now today is much more exciting because people used to think that the muscle is regulated by only the neurons. Today, we know that actually if you exercise, you produce what we call as the myokines, right? Small proteins, RNAs, and things. And those myokines goes to actually to your neurons and keep you happy. And it requires at least around 30 minutes of walking and running. So if you are running for 10 minutes, then the myokine will not release. What she has shown is that you need to, at least in the human condition, around 40 minutes. So if you start walking for 40 minutes, Mewasar will tell you that then you get the excitement, actually. You want to do more, right? So do you feel that? And that you can blame it to her. Today, what she has shown is because of the myokine produced from the muscles, it goes to the neurons, make it better. Okay, right? And Prasanna has been working extensively on the mitochondria, and he has now identified molecules which are expressed differentially in the different muscles. Okay, right? Yeah, thank you very much. Yeah. Yeah, it is there, sir. So the defective one has. Yeah, so there are many markers for the defective ones, which are like membrane potential, particularly if you look at, and they are recognized by multiple regulators again. So caspase 3, for example, can recognize all those things, and you can target. Yeah. Yeah. No. The genome, when we fuse, actually genome also fuse. Yeah. The whole, even the crystal, everything fused, remodel. That is why it is called as a remodel. That has been going on from long time. You have to recently, last 2 years, you have multiple papers on how those are recognized. Yeah, but you will have the multiple copy once it is used, right? And they are remodeled also. The mitochondrial remodeling is a very hot topic.
Sir, you said the removal of degraded mitochondria will help in extension of mitophagy.
Yeah.
So the presence of this degraded mitochondria, how is it actually functioning? Is it affecting the functional mitochondria or the cell?
It's just like, when we were young, when Mysore was not too many crowded, you didn't have garbage. Right now you have too many garbage. It's just like that. As you grow older, you have more A's, and the mitochondria, some of the mitochondria will be more in garbage, right? And if you recycle it by what we call as the not only mitophagy and autophagic recycling is the mitophagy, right? Yeah. So you recycle the garbage. Just like that, you recycle the dead mitochondria. That is where the cell can grow more space, and that is how they survive.
Okay, so it's not directly affecting any particular cell organelle inside the cell?
No. It of course it requires the lysosome to fuse with the mitochondria to make it as a automitosome. I mean, that autophagy, but there is another name for that. I mean, it's not after COVID, huh? Yeah. So after the COVID, I am suffering from dementia. So so things are not. I mean, words will not come very fast. So autophagosome kind of thing, but the autophagosome, mitophagosome, you get it. Completely now. Autophagy for the mitophagy is completely different name it is given, because mitophagy is involved in cancer. You look at it, for example, for the cancer cell, you need the autophagy to be upregulated, right? So that these are recycled, and the cancer cell use those enhances its survival. So it is a big thing now. That is where I'm interested in the aging. I mean, I'm diabetic. I want to live longer. I mean, I'm just looking at how to survive. And whatever drugs I will discover in fly, without telling other people, I will start taking. Right? Yeah. Okay. But mostly, I'm not joking, and I do that from the Ayurveda, because people say the Ayurveda is safe. That is where I'm now. If you look at, I'm working with the Ayurveda system, Ayurveda concept, and looking at some of these Ayurveda cocktail or the things. But I'm very particular. I look at their toxicity first and look at things, and that is the good thing about having modern organisms. I mean, just we come them with the thing. If they are not dead, they are walking around, the flies or the zebras and things, then I feel they're all there, right? Then you can start. Right? Yeah, that is what I do.
Thank you, Nishima.
Yeah.
So when exactly it will release?
So myokines are usually, once we start exercising, that is where the myokines start releasing. But what they have shown, my Mamta, they have got it. So what they have shown is that it takes roughly around 30 minutes to get for the myokine to reach the brain neuronal system. That is where you feel happy. So you need to run for 30-40.
Yeah, that's what.
Yeah, that's what I have. You need to run for 30. Yeah. Yeah, it's different.
There is a less energy.
Yeah.
Is there any role of mitochondria?
Yeah,
The regeneration of the mitochondria is reduced or what?
Not only regeneration. I mean, the moment you have diabetic, I mean, the cells are very happy and they die happily. I mean, they have lots of sugar, right? Yeah. So mitochondria, the cell death, everything is more in diabetes.
Okay. The increased sugar level reduces the regeneration of the mitochondria also?
Yes, that is where you have the glycomet. So you take the glycomet, you are making mitochondria to function more. It's just like, but remember, it's just like engine, no? You are pressurizing it more by thing, right? And one day it will come, I had enough, and it will exhaust. So those who are taking glycomet in the belly, the allopathic doctor will say that you can take up to 8 grams, right? Please take it from me. As a scientist, I will never say that your sugar is 650, you take 8 grams. I will not. Okay? Because you are poisoning. This has got a huge impact on your mitochondria and cells. So instead, I will tell them that you do other things, right? Yeah. And I'm practicing with Ayurveda, and today I can tell you that Chirayata, or right? Then is the powder. That works much better. Jiraya. You can get it from Amazon, Flipkart, Ayurveda Assistant. It's a small powder, 80 rupees per box, right? That will survive at least like 2 weeks. And I have tested it in everywhere. This is kind of antioxidant. You put half of the spoon in the one night. People have shouted at me for telling this thing, but these are my other experiment I'm telling you. Okay? Right? And you mix it and keep it overnight, drink in the morning. You can see the difference within 10 days. Your skin will start glowing, your inflammatory things which are associated with the diabetes, everything. Diabetic patients, you can identify it. You will set inflammatory, thing on the skin. Those will be gone in the Indian system is that, we said that, even the cow dung is good and things. But what we need is the proper science behind all. Proper science in the sense that toxicity, then, how they are affecting and things. And you will find the kind of science from us in next maybe 6 months' time. We have worked from pomegranate, and today we have identified the compound, but taking pomegranate is much better than taking the compound, okay? I can tell you that. And that compound is, we are able to cure, not cure, up to some extent, alleviate the, epilepsy. Yeah, right? This I haven't talked because there is lots of work going on in the lab. But this is what excites me. Okay? So if I go to lab and my student says that, sir, the compound goes to the brain, I was able to see it, I see and everything. That is where the difference is. Okay? So think about it. And please think about it, because he hasn't come up at the moment, but think about 2 pieces of advice. You see, every day when you get up in the morning, you say that you should be better than yesterday. So Upendra, I was yesterday. Upendra today should be better in terms of thinking. And you don't compete with somebody in your class or right? Yeah? Okay? You don't compete with anybody. Just compete with God. That is where you can grow. Right? If you compete with yourself. You can compete with yourself anytime. Even when you get older, your wisdom will be much better than yesterday. Even though your body will be worse than yesterday, but your wisdom will be more. Right? That is where you can make it. And we have only one life. And when you can make a difference, just please make it. Okay? Right? I'm done. Thank you very much. Yeah.
Personally and also on behalf of the organizers and University of Mysore, I would like to thank Professor Upendra for such a dynamic and information-laden lecture on mitochondrial dynamics. Thank you, sir. Or online idara nodi alli. Ondsati nodbidi. Ilva? Okay. We shall move on to the next session. Dr. Radhe S. Gupta is the next speaker. He is a professor in the Department of Biochemistry and Biomedical Science at McMaster University, Hamilton, Canada. He did his postdoctoral work at St. Louis, USA, Toronto. His main research interests are in using genome sequences to identify novel molecular markers that are useful for diagnostic, therapeutic, and evolutionary studies. His group also work in the field of mitochondria and heat shock proteins, comparative genomics, molecular genetics, and prokaryotes. On behalf of the organizers, University of Mysore, I cordially welcome him to deliver the lecture, but we are playing a recorded one. Shortly, he will be online.
Good morning, everyone. My name is Dr. R.S. Gupta. I'm a professor in the Department of Biochemistry at McMaster University, Canada. I thank the organizers for this conference for inviting me to speak on the occasion of superannuation of Dr. Ramachandra. I've known Dr. Ramachandra since mid-1980 when he spent 2 years as a postdoctoral fellow in my lab. Dr. Ramachandra was one of the most dedicated and capable researchers who have passed through my lab. And it was expected that he will become a leader in the field of genetics. Dr. Ramachandra's stay in my lab predated the genomic era. Hence, the work that I'll be speaking today has not yet begun, but its foundations were laid during that time. As the other speakers in this conference has indicated, genome sequences have revolutionized all areas of biology and health sciences. However, one area where genome sequences have a particularly dramatic impact relates to understanding the evolutionary relationships among microorganisms. So for the past 15, 20 years, research work in my lab has primarily focused on understanding the evolutionary relationships among microorganisms. So to begin with, let me give you an overview of the current understanding of evolutionary relationship among microorganisms. Now, the evolutionary relationship among microorganisms is commonly determined based on phylogenetic tree, and most of it is currently based on 16S or small subunit ribosomal RNA trees. But as the genome sequences have become available, it's also possible to construct tree based on large number of data sets of proteins or gene sequences. However, in all of the constructed phylogenetic trees, the different groups of organisms are found to form various clades. And in the case of prokaryotic organisms, about 30 to 40 different clades are seen in the phylogenetic trees. However, the trees have a number of limitations. One of them is that the branching order of the different organism or species, many clades in the trees are not reliably resolved. Second thing, based on the tree, it's difficult to reliably demarcate the boundaries of any given clade. And the third important limitation of the phylogenetic trees is that the trees, they do not provide any genetic or biochemical properties that is commonly shared by a given group of organisms. 5 years, the main focus of our work has been aimed at identifying, using genome sequences to identify novel molecular markers or characteristics that are specific for different groups of organisms. And in my lab, the work that we have done, we have pioneered the studies of discovery of two types of molecular markers. One of them we refer to as conserved signatures inserts or deletions, or CSIs, and the others are conserved signature proteins. And I will briefly, in the next few slides, I will briefly indicate what these two types of markers are and how they are useful for understanding, gaining information about different types of organisms. So this particular slide shows you the example of a conserved signature indel. In this slide or cartoon, basically what I have shown you is a partial sequence alignment of a protein. And in this region, this is a highly conserved region of the protein, and all of the dashes in the sequences in this slide, they indicate sequence identity with the amino acid on the top line. So what you observe here is that there are different species, X1, X2, or A1, A2 to X1, X2. They could be organisms at different phylogenetic level. They could be phylum, order, family, or species. Now, what you see here in this diagram is that all of the members of group X, they contain this large insert, which is shown in this box, whereas all other species or all other organisms, they lack this particular insert. And this insert is present in a conserved region, and it's flanked by, on both sides, by conserved region, indicating that it's a reliable characteristics. So just to indicate that the conserved indels that are useful for phylogenetic or biological studies, they are flanked on both sides with conserved regions. And these conserved regions ensures that the indel is reliable and it's not resulting from any kinds of artifactual alignment or other characteristics. These types of indels are present at different phylogenetic depths. They could be specific for phylum, they could be specific for order, genus, family, and even subspecies and substrains of species. And I will give you, in my talk, I will give examples of indels that are specific at different phylogenetic level. Now, the inferences based on these conserved indels are also generally not affected by other limitations of phylogenetic tree construction methods, such as lateral gene transfers. Now, these based on these CSIs, one can also root them based upon the absence or presence of these CSIs in the ancestral species. One can determine whether the insert in question, indel in question, is an insert or a deletion. If all the other outgroup species are lacking the indel, then the indel is an insert. Otherwise, if the outgroup species are containing the indel, then the indel is a deletion. So this is an example of indel. Now, these indels, they possess number of very important and useful characteristics. One of the properties is this constancy, that these are highly reliable characteristics, and they both small and large indels both are equally reliable. Here I'm showing you an example of an indel in the HSP60 protein that was identified in my lab in 1995. That's predating the sequencing of any genome. At that time, based on the available genome sequences, it was inferred that this indel is a shared properties of gram-negative bacteria and as well as eukaryotic organism, but it's absent in all other gram-positive bacteria. Now, over the years, large number of genome sequences have become available, and as every genome gets sequenced, if one can examine the presence or absence of these sequences in different genomes, and as I have indicated in the slide on the right, that these indels are highly reliable characteristics. So, for example, in the 2010, the number of genome sequences are increasing, but the expected presence of indel in the indicated group is highly correlated with the particular group. So, for example, all 1,500 species from at that time in 2010 contained this indel, whereas all of the species from this group did not contain, and one or two exceptions were because of gene duplication event. So these indels are highly reliable characteristics. Another important characteristics of these indels is that they are also essential for the group of species where they are found. For this particular indel, we have carried out extensive experimental work where we have either removed this indel by experimental means or made change the amino acid in this position by means of molecular methods. And again, any kind of change in this the insert position, either the absence or major change in this indel sequence, is incompatible for cellular growth, indicating that these inserts or deletions are playing important functions in the organisms where they are found. Now, this particular slide, it gives you an example of the conserved signature proteins. Now, unlike the indel, where the protein in question or the gene or protein in question is present in different organisms, these signature proteins are uniquely found in a particular group of organisms. And in this particular slide, I'm showing you the results of blast searches with a protein which is only found in Borrelia species. So in this case, all of the blast hits, significant blast hits, are for Borrelia species. And except for Borrelia species, there's no other significant blast hits are observed. So this protein or gene is entirely uniquely found in Borrelia species. Now, so in my talk, I will be describing different examples of usage of these conserved indels for understanding the evolutionary relationships among different organisms. And also I will be describing some applications of these conserved indels for a number of studies. So my talk today will focus on maybe three main areas. One is I would be looking at evolutionary relationships among mycobacteria and how our understanding of that has been advanced by genomic sequences. I will also be then looking at briefly at the evolutionary relationship and classification of species from the order Enterobacterial, which include E. coli and related species. And finally, I will discuss some potential applications of the identified molecular signatures for diagnostic and therapeutic developments. So let me begin with indicate to you the importance of Mycobacterium. So genus Mycobacterium contains more than 200 different species. And these species include Mycobacterium tuberculosis and leprae, which are the causative agent of tuberculosis and leprosis. However, most mycobacterial species are not pathogenic, and many of them are commonly found in water and soil. However, there are no reliable means existed until about a few years ago for distinguishing the pathogenic mycobacterial species from non-pathogenic mycobacterial species. And also there were very few molecular or genetic characteristics were known that were specific for the disease-causing organisms like Mycobacterium tuberculosis and leprae. So using available genome sequences for Mycobacterium species, we have carried out extensive phylogenetic and comparative genomic analysis. So this particular slide, this work which has been published in 2018. So in this particular work, we have, first of all, we have carried out what you call phylogenomic trees. And these trees were based on very large data sets of protein sequences. So basically, one of the trees is based on the 1040 core proteins that are commonly shared by all mycobacterial species. And likewise, the second protein is also based on 136 conserved proteins that are commonly shared by all actinobacteria. Now, you won't be able to see all the details of these trees, but basically the main point here is that in these phylogenetic trees, all the known mycobacterial species, they form 5 distinct clades. And these are very strongly supported clades. And these species from these clades are shown in different colors in this tree, for you to easily see that how they are distinguished from each other. And this particular diagram on the right, it shows a conceptual diagram indicating the main clades and names that we have given to them. Okay, so these are the 5 different clades of mycobacterial species that are seen in different phylogenetic tree. Now, the phylogenetic tree, as I mentioned previously, they do not tell you anything about what unique properties are shared by different groups of microorganisms. And also the branching order of the species in tree can often be variable. And because in most cases, it's difficult to construct trees such as the one that are shown on the right. In most cases, trees are based on 16S ribosomal RNA or other single genes, which are not very reliable. So our next task was to identify genome sequences to identify molecular markers that are specific for different clades of Mycobacterium species. So here in this diagram, I'm showing you some examples of molecular markers. So for example, in this case, on the right, the panel on the right, it shows a 6 amino acid insert, which is uniquely shared by all members of this abscessus chelonia clade. Okay, and this is just one example of the CSI that is specific for this clade. But in our work, we have identified 27 different CSI in different proteins, as well as 24 conserved signature proteins that are uniquely shared by all members of abscessus chelonia clade, but they are not present in any other Mycobacterium species. Likewise, the diagram below, it shows an indel which is specific for the Fortutum vacui clade. This particular group of Mycobacterium, they are also referred to as fast-growing species or fast-growing mycobacterial species. So again, this 5 amino acid insert is uniquely shared by all 46 members from this clade, but it's not present in any other bacteria. And again, we have identified multiple CSIs and CSPs that are specific for this clade of Mycobacterium. Now, in this diagram, I'm showing you other CSIs and CSPs which are specific for other clades of Mycobacteria. This is a terre clade, and again, we are describing to you, I'm describing here this particular CSI which is specific for terre clade. Multiple CSIs and CSPs have been identified, and this is the tuberculosis simian clade. This particular clade contains species such as Mycobacterium leprae and Mycobacterium tuberculosis, and these are slow growing Mycobacteria. And again, we have several CSIs which are uniquely found in the member of this clade but not present in other Mycobacterium species. So based upon large number of molecular signatures that are specific for each of these different clades of Mycobacteria, based on these markers, these clades can be identified not only in phylogenetic terms but in reliable molecular terms. And as I mentioned previously, these markers have high degree of predictive ability. So they are expected to be present in other members, other species from these clade as well. So based on these studies, what we have basically, this is showing you a summary of the results from these studies that we have carried out. So all of these different clades we can identify in molecular terms. So this work, based on this work, we have divided the genus Mycobacterium into 4 novel genera and an amended or a delimited genus Mycobacterium. So for example, the species from this other genera, they are now given by the different other clades, they are given new names. So the species from Abscessus chelono, they are now referred to by the new name Mycobacteroides. Species from the rapid growing clade, they are referred to as Mycolici bacterium. Species from the terre clade, they are referred to as Mycolici bacter. And only the species from the simian and tuberculosis clade, they are referred to now by the genus Mycobacterium. So genus Mycobacterium is now limited to only this group of species, which include tuberculosis, leprosy, and other related slow-growing Mycobacteria, which are generally disease-causing bacteria, and all species from these clades are known by these new names that we have given based on our work. So this work, it was published in 2018, and I have indicated this work has been highly impactful. There are already more than 190 citations to this work, indicating the importance of this new classification that we have proposed for the Mycobacterium species based on genomic analysis. Now here I want to describe, we have carried out some further work on members of the Mycobacterium clade. So as I mentioned, Mycobacterium clade, the delimited Mycobacterium clade, it contains Mycobacterium tuberculosis, leprosy, as well as it contains a number of other species. But still this clade is fairly large. And the Mycobacterium tuberculosis, as we know, is a single most infectious disease-causing organism. There are more than about 1.6 million deaths per year from this disease. And many of these strains of Mycobacterium tuberculosis, they have developed resistance to all available antibiotics. So there's a novel, and particularly in the context of India, the incidence of tuberculosis is fairly high in India. So there's an urgency to develop novel means for rapid detection of tuberculosis and tuberculosis, and also importance for developing novel means for treatment of drug-resistant form of tuberculosis. So we recently published another paper where we identify molecular markers, how genome sequences can lead to identification of potential targets for diagnostic and therapeutics of tuberculosis. So in this work, we have carried out again in detail further analysis of members of the tuberculosis clades. And the tuberculosis clade, like I mentioned, it again contains, the species from this clade again form multiple distinct other clades. So like these are species from the leprae clade, and there are other clades, simian clade, and different other clades that are part of this clade. Okay, but all of the species from the which cause tuberculosis, they are part of this tight complex referred to as the tuberculosis complex. So this includes species like Mycobacterium tuberculosis, Mycobacterium microti, and so on, and are very closely related species. So in this work, we have focused on identifying molecular signatures that are specific for the tuberculosis complex of bacteria, which are the main species responsible for tuberculosis causation. And I'm showing here two different CSIs in two important protein. Okay, one is this MRB protein, and again this is cis-H protein. So again, in both cases, these conserved inserts shown in the colored boxes, they are specific for the tuberculosis complex of species. All 9 members of the tuberculosis complex of species contain these inserts, whereas all other mycobacteria or other bacteria, none of them, they contain these CSIs. And these CSIs, as one can see, they are present in highly conserved regions. And so we have, as a result of our work, we have identified, in fact, a very large number of CSIs. In fact, we have identified 63 CSIs that are uniquely shared by members of the tuberculosis complex of bacteria. So these CSIs provide us novel tools for different types of studies that I will describe briefly later on. So now let me briefly move on to the uses or impact of genomic studies on clarifying the relationship among members of the order Enterobacterial, which include E. coli and related species. So here I've indicated in this summary diagram, okay, our understanding of the order Enterobacterial before 2016. That is before we undertook the study, our genomic studies on this. So before 2016, the order Enterobacterial, it contained a single family, Enterobacteriaceae, and within this, the E. coli is a part of this family. So as I've indicated, this order contained a single order, a single family, and within this, there were different types of species and many different genera. Okay, more than 50 genera are part within this family, and this contained different kinds of plant pathogens as well as animals and human pathogens. So animal pathogens include Salmonella, Yersinia, and of course E. coli, pathogenic E. coli. And so this was a highly diverse group of family or order. And it was difficult to there was no distinction made about how different large number of species that are present within this disorder are related to each other. So we undertook detailed studies similar to that I have described for the genus Mycobacterium, and these studies were published in 2016 in this paper that I have described here. And in this case, our work identified a large number of CSIs which are specific for the order Enterobacterial. So 5 of these CSIs were commonly shared by all Enterobacterial, whereas large number of other CSIs, okay, they were specific for different clades of within Enterobacterial. And all of these clades which are shown here, they are reliably distinguished in genome-based phylogenetic trees, which are not shown here. So these clades are distinct based on their phylogenetic branching pattern. But then, importantly, we have identified large number of molecular signatures which are unique to this group of these particular clades of species, enabling their identification in molecular term. So based upon these studies, our work led to division of the order Enterobacterials into 7 families. Okay, so the family Enterobacteriaceae was reduced to a small number of this particular group, whereas all the other Enterobacterial species were divided into these families. And again, our classification is now widely established, broadly established. And as one can see, since 2016, this paper has been cited 200 and almost 300 times, and it's one of the most highly cited paper in this particular journal and in this particular area. Okay, so leading to the importance of the work that we carried out in this regard. Now, we have further carried out work on Enterobacteriaceae family. So like I mentioned, these were the 6-7 family that we initially divided the order Enterobacterial was divided into, but then now we have focused the further work on Enterobacteriaceae family, and further analysis of this family indicated that this family consisted of at least 6 different clades, okay, which we have indicated as here by different names, and each of these clades could be identified multiple CSIs, and these are subfamily level clades. Okay, although we have not proposed a formal classification of these clades into subfamily, but this possibly will be occurring in the coming months or year. Okay, but the point is that all of this we can now clearly distinguish these different clades of bacteria in both phylogenetic terms and molecular terms. Now, I want to describe some further work about the Escherichia clade. It contains E. coli as well as number of related species, and I have indicated here one CSI which is specific for the Escherichia clade. So Escherichia clades include species members of the genus Escherichia, Citrobacter, Salmonella, the 4 genera that are part of this particular clade. And so we have identified large number of CSIs which are specific for this clade. And then within this clade, we have carried out further work where we have looked for CSI specific for either different genera that are part of Escherichia clade, as well as the figure on the right shows a CSI with members of the Escherichia genus. They contains this particular CSI, but this CSI is not present in other members of the clade. So again, based on the CSIs, one can in molecular terms, we can identify the order Enterobacterial, we can identify the family Enterobacteriaceae, we can identify this subfamily which we refer to as Escherichia clade, as well as we can identify the genus E. coli Escherichia in molecular terms. Now, this approach that I have described for identification of molecular signatures, this is not limited only to the prokaryotic organism or microorganism, but this can also be used for identification of signatures specific for different eukaryotic organisms. And I won't have time to go into it, but based on using the same approach, our work has provided important insights into the evolutionary relationship among higher organism. But like, for example, in this particular we identified large number of signatures that are specific for vertebrates species and how vertebrate species are related to other members of the phylum Chordata. And in other studies, we have looked at molecular signature specific for the Plasmodium as well as nematode species as well. So now towards the end, I will move over briefly to the applications of the discovered CSIs and CSPs. I have indicated that CSIs and CSPs, they allow demarcation of different groups in molecular terms, and they have proven very useful for evolutionary and classification studies. And the properties of the CSI that I have indicated is that they exhibit high degree of specificity for the indicated group of organisms. They also indicate high degree of predictive ability that other organisms from these groups will also contain these CSIs, even if though their sequences are not available or those such organisms have not been discovered. So because of these properties, the predictive ability and specificity, these CSIs and CSPs, they provide important means for development of novel diagnostics and therapeutics. Furthermore, like I mentioned, all of these CSIs are present in important proteins, and if they changes, and I've also indicated that these CSIs are important, they play an essential role in the given organisms. So although the functions of these CSIs are not known, but it's expected as further genetic and biochemical studies are carried out on these molecular or genetic characteristics, it would lead to discovery of novel properties that are specific for different groups of microorganism or organisms. Now here I have indicated how these CSIs and CSPs can be used for diagnostic development of novel diagnostic assay. So for example, if you have a CSP, which is specific for different given group of organisms, one can design PCR primers based on it. And this PCR primer will only amplify, give a positive result for the given group of organism. All other groups of organism, because they do not contain this gene for the CSP, you should not see any positive results. So they provide important means for development of highly specific diagnostic assay. Likewise, in the case of CSIs, okay, where the gene may be present in other organisms as well, one can design PCR primers that are flanking the conserved the CSI based on conserved regions that are flanking. And in this case, based on the size differences between the amplified between the fragment that are amplified, right? One can again distinguish whether the organism, whether the amplification is because of the presence of the indicated group of organisms or not. We have carried out as a proof or principle for this approach. We have used the CSI approach for development of a highly specific assay for Bacillus anthracis. And this work was carried out in collaboration with a group in Public Health England or Health Protection Agency in UK. And in this, the species Bacillus anthracis is based on 16S ribosomal RNA. Its sequence is identical to that of the Bacillus cereus. So very difficult to distinguish these two groups of organisms based on 16S ribosomal RNA. So in this particular study, which was, like I mentioned, was carried out in collaboration, we identified a large indel, okay, in fact, a 24 amino acid indel or 72 base pair indel, which was specific for Bacillus anthracis. And the specificity of this indel for Bacillus anthracis was determined based on examination of large number of available strains. So it's a highly specific deletion which is found only in Bacillus anthracis and not present in any other Bacillus species. So based on this large indel, we developed both a PCR, conventional PCR-based assay. Because of the large indel, one can easily distinguish the Bacillus cereus, which leads to amplification of a larger fragment as opposed to Bacillus anthracis species. At the same time, we also developed a rapid pyrosequencing assay, whereby large number of clones can be, or large number of samples can be at the same time within a short time. They can be determined, they can be examined whether they correspond to Bacillus anthracis or Bacillus cereus. Now, this is another example of the use of conserved indels for the detection of organisms. So like I mentioned, there are certain groups of E. coli which are highly pathogenic, and particularly E. coli 0157H7 is responsible for many outbreaks of gastrointestinal diseases. And this is a very important concern because these species can be contaminating water resources or other agricultural food products, right? And so this leads to a major problem. So in this case, we identified a conserved insert which was uniquely present in the, which was specific for this particular clade of organisms. And it's indicated here that this 3 amino acid insert in a highly conserved region was only found in the 0157H7 related strains, and all different strains were examined to establish its specificity. And whereas all other E. coli's do not contain this particular insert. And so based on this, we have developed a PCR TaqMan probe, okay, for quantitative PCR analysis, and this and the specificity of probe and the usefulness of this probe was examined for looking at the detection of this 0157H7 in freshwater environment, and it was shown to be highly specific and it can detect the presence of this pathogenic organism in water sample at a fairly low concentration. Now, I mentioned that the CSIs can also be used for therapeutic purpose, for development of novel therapeutics. And the usefulness of these CSIs as potential therapeutic targets is based on at least three main observations. First of all, these CSIs, as I mentioned, they are highly specific for a given group of organisms. And in most cases, like I mentioned, these are not present in the human homolog because they are present for disease, a particular protein only in a given group of organisms. And we have shown that the human homologs of these proteins, they lack the CSI. Okay, we have also shown that these CSIs, in the number of proteins that we have examined, that they play essential role there. So whatever function they are carried out, it is essential for the CSI containing organism. And the third thing is that these CSIs are present as surface exposed patches on protein surfaces. Like this is an example of where this particular CSI, which is specific for mycobacteria and actinobacteria, is located on the surface of the protein. And so these surface exposed loops or patches, they are involved in enabling novel protein-protein or protein-ligand interaction that are essential for this group of organisms. So the prediction is that if one carries out chemical screening of small molecules to find small molecules that bind specifically to this CSI, which is the surface exposed CSI, then in these cases, so this is showing you the approach. So this is the CSI containing region, and we can have we can screen small molecule chemical libraries, okay, which could contain millions of compounds, and we can look for compound that may bind specifically to the CSI region. And it's expected that many of these compounds which bind specifically for the CSI, they will prevent or they will interfere with the functioning of the CSI in cellular function. And yes, the function of the CSI are essential. So these compounds binding to the CSI will be exhibiting growth inhibitory properties toward this organism. So we have identified large number of compounds that are directed towards the CSI, and we are testing whether these compounds exhibit antimicrobial activity or not. So in summary, what I have indicated so far is that what we have carried out using genome sequences, we have identified large number of CSIs and CSPs that are specific for different groups of organisms. And because of that, we can now reliably identify these groups in molecular terms, not only in phylogenetic terms, but molecular terms. Okay, and these molecular terms, and further, I have indicated that these CSIs and CSPs exhibit high degree of specificity and predictive ability, and based on them, one can develop different kinds of novel diagnostics as well as potential drug targets. And lastly, because these compounds, these CSIs and CSPs, they are essential for these organisms, they play important role in these organisms. So we expect that further genetic and biochemical studies on these CSIs and CSPs should lead to discovery of many novel properties that distinguish different groups of organisms from each other. Now, here in this particular slide, I have indicated some of the individuals from my lab who have worked in my lab over the years. And these are, in fact, interesting to show that this is Sharat Ramachandra, Dr. Ramachandra's son, who also spent a few months in my lab when Dr. Ramachandra was a postdoctoral fellow in my lab. So my association with Dr. Ramachandra goes back very long time. So lastly, I end my talk by wishing Dr. Ramachandra my heartiest congratulations on reaching this important milestone, and wish him good health and great success in his various leadership roles in the fields of genetics and genome.
lecture on the impact of genomics on discovering novel molecular characteristics, specifically with respect to the tuberculosis disease. How the phylogenomics and comparative genomics studies indeed can be used as a tool, and using that information in a new perspective, how one will be able to utilize the knowledge that we are going to gain from the phylogenetic analysis with respect to the insertion and deletions in the very specific areas. We will be able to use those proteins as targets, and then develop novel lines of drugs. This was indeed a very interesting lecture, and we will convey our sincere gratitude to Professor via email. And next we shall move on to the next lecture of Professor Rai from Banaras Hindu University. And I hand over the session to Professor Shakuntala.
Good morning, all.
By the way, this is the program, right? Program is that as a person, for the DKNO, I know it from now. Like when I was first year, I used to come and attend all our Prof.N.B. He's a very nice guy. He speaks very well.
But he's unable to lock in, right? This is the generation gap. I mean, when it comes to actually putting
Upendra, his guide is better than him. Lakotia sir.
Because
He's better than him. He's more. He's now 80.
Lakhotia sir is attending most of the meetings, no? So he's expert.
More advanced.
I think he should be in. Okay, somebody has got the answer for my questions. Yeah, today's question. Yesterday you didn't answer. GATE and CSIR, I mean, many times I don't believe in the things because we find, we interview the CSIR rank number 1 in the midterm admission this year. That was January 1st week. I mean, she didn't answer anything, so I believe it in the number and the rank. I mean, yeah, I never passed CSIR exam actually. I appeared, yeah. That time they used to select 162, but I got SRF. When I went to Delhi and I get the interview, then I'm good in the, up to some extent, the interview. Interview is okay, I mean, you can explain them. In the questions, you can't explain them, that is the problem, right? Yeah. No, I mean, believe me, so you got answer for yesterday? Yesterday's Mendel's this thing, you see, is very easy. So in genetic system, somebody yesterday also talked about the modifier. So there could be second site mutations, right, in the short plant, broad plant, second site mutations which can suppress the tall plant. That's it. These are called as the suppressor. Okay, and if some mutation, second site mutation, which will enhance the phenotype, these are called as the enhancer mutations, right? Okay, so modifier effect, you study it, but you will not apply that if somebody asks you the question, right? Okay, so think about it, just like that. You got it? Yes? Yeah, it was very simple answer. Remember.
We can also continue with the question answer session if they have some mitochondria talk questions on the answer.
Yeah, today's question, I would like to see somebody answering.
Very short it was.
If you don't have the translation in RNA state, but if you can translate directly from the DNA, what will be the advantage and what will be the disadvantage? But I told you, intron is only in the eukaryote, but think about the prokaryote. You don't have intron. Maybe he can send his slide and I can give it.
Yeah, you can stand up.
No, I know. I mean, his work is on the rec one and things, so I have collaborated with him. So if he can send the slides, I can give the talk.
Sir, we can adjourn for tea if possible, if it takes time. Okay.
Raghunath sir, can I ask some questions? Yeah. Well,
Good morning, sir. J.K. Roy, Professor J.K. Roy. Can you hear me? Professor J.K. Roy? ಅವರು ಇದು ವಿಡಿಯೋ ಆನ್ ಮಾಡ್ಕೋಬೇಕು. ಆಡಿಯೋ ಅದೇ ಅವ್ರದ್ದು ಆಡಿಯೋ ಆನ್ ಮಾಡ್ಕೋಬೇಕು. ಪ್ರೊಫೆಸರ್ J.K. Roy, good morning. Can you hear me?
he has to unmute.
Professor J.K. Roy, good morning.
Yes.
Good morning.
Good morning. Good morning.
Okay, so you can hear me, right?
Yes.
Yeah, we welcome to the session, sir.
Thank you.
Okay, so good morning all. Once again, we are here to hear the lecture of Professor J.K. Roy. And J.K. Roy is a senior professor in the Department of Zoology, Banaras Hindu University, Varanasi. Professor Roy got his PhD under S.C. Lakotia, Professor S.C. Lakotia, and from BHU itself. His research interest lies in the field of Drosophila genetics, mainly understand the role of a small protein, G protein or RAB11, and he has published many interesting papers on this. And interaction of viral gene and some of the host genes are in progress to decipher the initiation and aggravation of the cervix cancer, and his work on cervix cancer is also very highly interesting, and many papers has been published on this aspect. And he got DST Young Scientist and award in the year 1993. And Professor Roy is life member and ex-president of the Indian Society of Cell Biology, and is also a life member of Indian Society of Developmental Biology, and is a member of Board of Studies of many universities. And apart from this, is a very humble and humane person, and highly active, and he has participated and organized many conferences, and we also attended many conferences organized by him, and meticulously he does his job, and very nice person. And also he apart from this, he received many awards, and the highly interactive person. And with this, I welcome him for this session, and over to you, Professor Roy. Professor Roy,
Thank you.
you can continue with the presentation, sir.
Yes. Are you able to see me?
Yes, sir. Yes, sir.
Okay. Thank
We can
you.
hear you very well, and we can see you, sir.
Okay. Thank you. Thank you. I can see Professor Ranganath. Sir, good morning. Good morning to all of you, and especially Professor Ramachandra. It is very nice occasion, and because Professor Ramachandra, he developed the Drosophila genetics in his lab, and especially I'm very happy that Professor Ramachandra many of his students, they are doing very well. So I congratulate Professor Ramachandra on this occasion, because he completed his regular course of service. I am sure, Professor Ramachandra, you will be there in academics for a very long time, and you will produce many more good students. So I come to the topic
of our research, that is Rab11 in epithelial morphogenesis. Initially, it was very interesting for us to reach to Rab11 because this is a kind of talk when you have retired. So it is very good that I take a bit history of Rab11, how we got involved into it. We were doing P element mutagenesis. And while doing P element mutagenesis, we found one of the very interesting phenotype, that is eye degeneration. Eye degeneration means a lot of fusion, lot of roughness, and eyes are sort of burnt kind of a situation. We got interested into this. We thought that some of the gene might be doing this. We tried to clone this gene. Because this was P element mutagenesis, we did plasmid rescue and we cloned this gene. When we sequenced and matched with the database, we found that this is Rab11. That is how we entered into the Rab11. I did say Rab11 is involved in eye degeneration phenotype. This was essentially due to, this was our earlier work, that this was due to epithelial morphogenesis problem. So let me come to the next slide. Yeah. So this is a slide where this is showing the importance of the Drosophila. If we compare with human has digestive tract, Drosophila human has a nervous system, the fly also has, body organization is also very definite. Circulation, excretion, skeleton, muscle, everything is there in the fly. When we come to epithelial morphogenesis, that is modification of individual properties of cells affecting tissue architecture. You can see there are cell intercalations, there are rotations, there are invaginations, there are sheet extensions, there are tube formation, the tube constriction and elongation, and so on. So these are under epithelial morphogenesis. And during gastrulation, during different stages, lots of cell changes occur. I'll come to that. Yeah. This is showing sheet elongation. Let us imagine this is a wound created. And then what happens? That the cells, they have to get extended to cover the gap. This is in addition to if some cells are dividing, so that is different. But here we are talking about the expansion of cell, then they come together, then they finally close the gap. Lots of adhesion molecules and polarity complex proteins define the differentiated state of an epithelial cell. This is a differentiated epithelial cell. Here, this is apical site, the top, the bottom is basal site, this side is basolateral site. So here you have lot of adhesion junctions. This is basolateral junction. And some of the polarity molecules like scribbled, DLG, LG, and so on. So in a differentiated cell, there are adhesion molecules and polarity complex protein they define the final polarity of a cell. There are different kinds of polarity. This is planar polarity. This is apical basal polarity. Spindle orientation is also depending on this polarity. This is front rear polarity, the top and bottom. There is another event, that epithelial mesenchymal transition, that also occurs many a times, that an epithelial cell becomes mesenchymal or a mesenchymal cells become epithelial. So these kind of properties are there. So who regulates the cell morphogenesis or cell morphology or cell? So this is the nucleus and this is endoplasmic reticulum. These are Golgi. So some of the vesicles from endoplasmic reticulum fuse with Golgi. Then some of the vesicles, they come out from Golgi and fuse with the plasma membrane. So these vesicles essentially make plasma membrane to grow. If plasma membrane has to be shortened, then more and more vesicles come out from plasma membrane, go inward. That is how plasma membrane shortening occurs. So essentially, during cell division or during any such kind of cell extensions, it is the plasma membrane that has to grow, and this is due to fusion of these vesicles that is coming. This is the same picture which is showing, this is endoplasmic reticulum through Golgi. Then you have some of the secretory vesicles and so on, they can fuse. So that is how the membrane morphogenesis occurs. If we see some of the proteins like RAS family protein, in RAS family you have the RAS itself, you have the Rho, RAC, you have RAB, you have RAN, and you have SAR1. So these kind of molecules are there. The RAPs are small RAS-like GTPases which regulate directional vesicular trafficking to and fro the various components of endomembrane system. So RAPs are very important component. As I said initially, that we reached from a phenotype in Drosophila, we reached to RAPs. There are different RAPs. Some of them, they are involved in certain path of the vesicle trafficking. Some are involved in inward vesicle trafficking. Some are involved in recycling kind of vesicle trafficking. RAB11 is involved in recycling. That is, this is going from this side. Some of the vesicles, they have come from this side, and these vesicles are being recycled and finally reaching to the membrane. So there you have role of RAB11, many other RABs. Here, when we are talking, wound healing is a process that requires coordinated cell shape changes, activated by cytoskeletal remodeling, cell adhesion dynamics, and morphogen-induced cell signaling. So this is for wound healing. In Drosophila, you have a very good same wound healing kind of a model, or this can be used as a wound healing, and that is dorsal closure. When you have the embryo, so essentially, initially embryo develops, gastrulation occurs. So when gastrulation occurs, the dorsal side becomes open. Initially, this is closed. The head is very close to the tail. That is, the head segment and the tail segment or the last abdominal segment, they are very close. When the embryo becomes straight, then that time the dorsal side gets opened up. So this is showing the dorsal open. This is dorsal open. And inside you can see the amniocerosa. Then these cells which are here, these are leading edge cells, or these are the lateral epidermal cells. So this is lateral epidermis, a leading edge, and these are the lateral epidermal cells. So they essentially change the shape and they finally close this. So that is how this happens. So this is the dorsal closure happened. This is due to the cell morphology changes. So Drosophila embryonic dorsal closure, and ideal model to study the underlying genetics of cellular processes governing wound healing. So this wound healing can be equated with this. Rab11 expression in drosophila embryonic epithelium is functional relevance. So where is Rab11 expression? Rab11 expression is in the this is coracle and this is Rab11. So this is the membrane marker and this is the Rab11. So Rab11 essentially is expressed throughout the cytoplasm, but close to the membrane also very much. When the mutation is there, so normally this is the dorsal side, this is the epithelial cell structure, and during dorsal closure, these epithelial cells gets elongated. So this is the elongated kind of a situation. When there is Rab11 mutation, what happens? These dorsal side, I'll come to the next picture. No, not next. I'm showing it here that these cells which get extended, when it is Rab11 mutant, these cells do not get extended. I'll come to more figures later. All of you, this audience knows the binary Gal4 US system of targeted gene expression in Drosophila. We can express any gene in a given region by this. And this is US Gal4 system, where Gal4 is a transcription factor. It is binding with US and activating a gene. Using this, targeted knockout of Rab11 was done from the dorsal epithelium. This results in dorsal open phenotype. So I was showing dorsal closure. This dorsal closure does not occur. And this resembles a JNK overexpression mutant phenotype. That means JNK, with all probability, this is involved with this. So this is targeted knockout of Rab11 from the dorsal epithelium, results in dorsal open phenotype. This is dorsal open phenotype. This is dorsal open phenotype. And this is the embryo gets turned like this. This is called puckering. So this resembles puckered E69. This is a JNK over. This is a JNK mutant. So what I was showing, I was trying to tell you that during dorsal closure, the cells get elongated like this. And that is how the gap is closed. The cells get elongated like this. This is the same picture merged, and this is Rab11 expression. In the mutant, this is Rab11 dominant negative mutant. This was driven by PNR Gal4, and this is US Rab11. So in this case, see, this is these are the cells here. This could not change the morphology, and because so it could not close the gap. So this is the measurement of the aspect ratio. That here you can see that there is difference in aspect ratio, that these cells are elongated, these cells are not elongated. Now, I take it, a targeted knockout of RAB11 from the DLE's result, this is dorsolateral epithelial cells, results in an ectopic caspase expression. So what happens? The question is, I'm sure you can see the question, that whether the cells undergo cell death. So this is these are the dorsal open phenotype, and this is a targeted knockout of RAB11. So this is RAB11 knocked out, and this results in an ectopic caspase expression. I'll show caspase expression. So this is caspase expression. This is caspase expression in the membrane region. This is caspase, same thing. So that means what happens? So in the membrane, you have very high caspase. In the cytoplasm, you have low. So essentially, this is cell death is occurring because of this. So if cell death is occurring, that means that pathways, that means the apoptotic pathways are on. So targeted expression of RAB11 loss of function mutation in the DLEs result in significant ectopic expression of pro-apoptotic gene HIT. This is HIT. This is in the normal condition. This is the situation. This is an embryo undergoing dorsal closure. So you have very less HIT expression. Here in the mutant, this is RAB11 dominant negative. You have a good amount of HIT expression. That means apoptotic pathways are on. So this is loss of function mutant of RAB11, also showed dorsal closure defects and activation of DRICE, that is caspase 3 in the lateral epidermis, much like the conditional loss of function of mutant RAB11. So this is amorphic loss of function mutant, that is, this is severe mutant. There also you have the dorsal closure defect, and this is showing the caspase 3 expression in a very high amount. For any such kind of thing, cortical cytoskeleton plays very important role. So cortical cytoskeletal specific myosin, that is myosin 5, physically interacts with RAB11. So this is there in literature that this physically interacts with RAB11. Here, what we have done, disruption of actomyosin organization in the dorsolateral epithelial cells was observed in RAB11 loss of function condition. So this is myosin, this is the situation, this is normal condition, and this is in the mutant condition. That actomyosin organization is lost. I'll skip this. So epithelial actin. Here we are trying to show the epithelial actin also. Epithelial actin organization in epithelial cells is regulated by cadherin-catenin complex. This is actin belt. This is epithelial actin. And this is organized by the cadherin. So this is actin. This is organized by cadherin. Now RAB11 has maintained epithelial crumbs and adherent junction in Drosophila embryo ectoderm. How does RAB11 mutation affect the epithelial actin organization? That is our question. What we are showing, RAB11 loss of function mutants show a disrupted and elevated expression of armadillo that is beta-catenin, in dorsolateral epithelial cells, which absolutely co-localizes with caspase-3. So what happened? Rab11 loss of function. So this is Rab11 loss of function. Here, this is showing elevated expression of armadillo. This is beta-actin, which co-localizes with caspase-3. So this is co-localizing with caspase-3. Here we are showing this is co-localizing with caspase-3. So this is armadillo. This is armadillo expression, and this is caspase-3 expression. This is merged. That is, this is co-localizing. So the question is downregulation of caspase. So that means downregulation of caspase in Rab11 loss of function mutants may result in a reduction of armadillo cleavage, and in turn may result in a rescue of embryonic lethality phenotype. So the question we asked, this is the result. The question we asked, can a blockage of caspase function? We are showing caspase function is elevated. The question is, just a minute. The question is, can a blockage of caspase function result in restoration of armadillo expression in Rab11 mutants? We showed that caspase-3 function is elevated. We want to now block the caspase function. When we did this, this is armadillo, this is down regulation of caspase in Rab11 loss of function mutant may result in a reduction in armadillo cleavage, and in turn may result in rescue of embryonic lethal phenotype. So what we are showing that it rescues the embryonic lethal phenotype. So that means essentially it is caspase Rab11 loss of function, where you have the cell dorsal closure is not happening. This is caspase mediated. So the next question is, does loss of cytoskeletal organization and dismantling of actin anchorage molecules result in induction of epithelial mesenchymal transition? What we are showing here, that there is one marker, MMP1. This is a marker of mesenchymal cells. Here what we are showing, that the mesenchymal cells marker, that is MMP1, is expressed. Rho 1, a component of, so MMP1 is a marker of mesenchymal cells. What we are showing that MMP1 marker is expressed. So Rho 1 is expressed, MMP1 is expressed. This is MMP1. This is MMP1 in normal condition. That MMP1 is not expressed. So this is the normal condition. And here you can see in the Rab11 mutant, the MMP1 is expressed. That means what happened? These cells, which were essentially the epithelial cells, now this is changing its fate, and this is sort of becoming mesenchymal cells. Do we have some more time, or excuse me? So let me go to this, that, am I audible? Hello? Am I audible to you? Am I audible?
J.K. Roy?
Am I audible?
Yes, sir, you are audible very much. And you
Okay.
have 5 minutes time.
How much?
5 minutes.
Okay, okay, okay. So then we are looking for another thing, that wingless. This is a molecule. So Rab11, so we are showing that Rab11 interacts with wingless in the regulation of caspase expression. So the question is how caspase is expressed. So here caspase expression, this is the canonical wingless Wnt pathway, and this is non-canonical pathway. In both, essentially what happens, that if this pathway is not going properly, then the caspase expression occurs. So caspase expression is wingless mediated, that is in the report. So can wingless disrupt targeted overexpression of wingless? What happens? So this is a Rab11 mutant, and this is wingless RNAi. This is wingless mutant. Does Rab11 interact with wingless in regulation of caspase expression? This is the question. This is caspase 3 expression, and this is Fas3. So this is showing partially rescued Rab11 mutant in first instar larvae, knocked down for wingless. So what we were showing? We were showing that wingless expression is high when you have Rab11 mutant. So now wingless is knocked down, and result is partial rescue. So this is the Rab11 mutant and wingless RNAi. So this is US Rab11 and US wingless RNAi. So here there is partial rescue. So that is the situation, that wingless mutant. So that means we try to look for, so do Rab11 mutants manifest their phenotype by affecting JNPK DPP pathway? We did show that yes. I think I'll cut short, and so I'll come to this, that summary, that Rab11 is essential for coordinated cell shape changes in the dorsolateral epithelial cells. In the process of dorsal epithelial morphogenesis, Rab11 could be affecting here I cannot see this. Maybe you can see it better. I'm not able to see this. Then perturbation of knockout of Rab11 function from dorsolateral epithelial cells during, just a minute. I'm not able to read this because of picture. So essentially what we try to look for, Rab11 function, and what we have shown that the Rab11, when it is not functional, result is elevation of caspase. When we take that caspase mutant along with Rab11 mutant, the phenotype is rescued to some extent. That means essentially this is caspase mediated. Same way we try to look for that when wingless mutant is there, when we combine wingless mutant with Rab11, the phenotype is rescued. So that means that these are the molecules which are actually interacting, and I think I'll stop here. I'll not go further. I'll stop here. That Rab11 has a functional role in membrane morphogenesis. Thank you. Thank you, madam.
Thank you, Professor J.K. Roy.
Yeah.
Can you we'll have a question answers.
Yes, yes.
Okay. Any students can have a questions because they do work on Drosophila.
Dr. Roy,
Hello?
Dr. Roy,
Yes.
thank you for your presentations.
This is Professor Ramachandra speaking.
Yeah, speaking, Ramachandra speaking.
Yes, yes, yes.
You brought to our younger generation students also. Thank you for your nice presentation.
Thank you. Thank you so much. And it is very, very nice that I could talk on this occasion. Yes, yes, yes. I could see you, and it is very, very nice. And I'm very happy to be part of this.
Yeah, we are very happy to include our the BHU group for this occasion also.
Yes,
Right?
certainly, certainly, certainly.
Thank you. Thank you.
And I pray God for your very long, active academic life.
I need good blessings now because I'm in the last evenings of my life.
Yes,
Right?
yes. And you should enjoy academically very well.
Thank you. Thank you.
This is our prayer.
So, Professor Roy, we thank you profusely for a wonderful lecture on RAB11 protein, epithelial morphogenesis, and its involvement in interaction with the process, cascases, and so on. Very nicely presented with good confocal illustrations. So, it's a model to our students also. Thank you very much on behalf of our organizers, on behalf of the university, and on my behalf. And I'll profusely thank you for a good lecture and giving time for you to present your work. Thank you very much.
We will now disperse for a short tea break. So I request everyone to assemble back in the auditorium in 10 minutes. So we'll start the next talk by 11:45. Thank you.
Dr. Shashidhar, please unmute your mic.
Yes. Yeah, I thought I'll do it when I'm ready, or when you are ready to hear me.
Good morning, sir. Good morning.
Hello, NPR. Good to see you.
Welcome to the session, sir.
Yeah.
Okay. So, Dr. L.S. Shashidhar, distinguished visiting professor of biology at the Ashoka University, Sonipat, Haryana. Professor Shashidhara received his PhD from University of Cambridge, UK. He works in the field of general biology, Drosophila, but also works with other insects. After a brief stint at NCBS, Bangalore, as a visiting fellow, he joined CCMB Hyderabad as a scientist and group leader. Following that, he moved to IISER Pune as a professor and chair of biology and has contributed to the growth of IISER Pune since its inception. So we have also seen the campus, and he has done a lot of things in IISER. Professor Shashidhara is an elected associate member of EMBO and is also recognized with many awards, including CSIR, Technology and SS Bhatnagar Prizes and JC Bose National Fellowship. He is elected fellow of all three academies of India. He has been elected as Vice President of INSA and Secretary General of International Union of Biological Sciences in recognition of his ability to understand the intricacies of the science administration. He has put many administrative posts in India and policy and its impact. In addition, he is also serving as a member of various apex committees of government and science and technology organizations. So I profusely welcome you, sir, for this session and for your time for the session. So thanking you, and over to you, sir. Professor Shashidhar, over to you, sir.
Okay, thank you very much for this kind and pretty longish kind of an introduction. You must have downloaded from the internet. I hope much of it I deserve. Anyway, it's a pleasure to be here today. Congratulations to Dr. N.B. Ramachandra and good friend of mine, and really respect the kind of work that he has done in contributing to both research and teaching in India, and in particular in the Mysore University, and training very large number of very good students. Some of the students have worked in my lab also, and I know the quality of the training has been pretty good. And also impressed that NPR has moved, seamlessly between Drosophila genetics and human genetics, and human genomics, all three aspects: genetics, applied genetics, human genetics, and human genomics. Not many people can handle, such diverse areas. Interconnection, of course, is genetics, Mendelian genetics, and I'm sure the students of zoology department, Mysore, which is another pioneering, place in the country for introducing not only Drosophila genetics in research and teaching, and in general, teaching classical genetics. And very often we think, modern biotechnology and genetic engineering methods have made classical genetics obsolete, but it is not true. Even today, much of the biological information can be generated only using classical genetics approaches. It's simply because every time when we are born, we are inheriting, genetic factors. It could be whole genome, it could be a mutated genome, it could be a variants of our genomic sequences. But still, the classical genetics, what Mendel discovered more than 150 years ago, and which was rediscovered subsequently, 50 years later, and the work of the Fly people who actually connected classical genetics to molecular biology. Because unless Thomas Hunt Morgan had shown that the chromosomes are indeed the carriers of the genes, or ultimately, finally, it was discovered that chromosomes themselves are the genes, genes attached one to each other in a string. And that basically led to all the revolution in molecular biology that we are experiencing today, right? So what I'll try to do is not to give a very detailed introduction about the kind of work I do. I thought I'll bring in some of the classical genetic approaches that we use rather than, trying to talk about the results from the lab. I'll sort of aim this talk at the student level, and all the senior people who are here, please, excuse me if the talk is too sort of, at an introductory level. So let me start sharing my slides. So in the footsteps of N.B. Ramachandra, I'm also sort of trying to move from applied genetics to cancer genomics and see, how we have moved and, what kind of work that we can do. It's a very exciting phase in my research career, the kind of work that we initiated in the last few years on cancer. So, like all developmental biologists, the question that we ask in the lab is how from a unicellular embryo you get multicellular complex organism, right? And we use Drosophila as a model system, and like NBR, I'm sure audience, although it's from the zoology department, most of you are familiar with the Drosophila as a model system. More specifically, the interest in the lab is two, right? How organs are positioned along the body axis. Along the anterior posterior axis, whether a human or flies, we have these very different organs positioned along the body axis. We have, eyes or, ears or, very different appendages. In Drosophila, the flight appendages, wing, there are only one pair of wings because they have dipterans, unlike lepidopterans or hymenopterans or honeybees or butterflies and moths. There's only one pair of wings, and these the second pair, which is supposed to be in the hind wing, the pair of hind wing, which is supposed to be on the third thoracic segment, are modified as small balancing organs, what we call as halter. And Ed Lewis and his colleagues and many other workers across the world worked on this, and they showed that it is the Hox protein, UBX, which is responsible for this. UBX is expressed in T3. If you remove UBX, you get wing to halter to wing transformation. That means T3 becomes T2. Or if you overexpress UBX, what is called gain of function mutation, you get wing to halter transformation, right? Complete transformation of T2 to T3, the fly would be lethal, wouldn't come out of the pupal case. So you don't see an adult fly with complete transformation of the thoracic T2 to T3. But what you can get is an appendage, right? As you know, all appendages are accessory and not necessarily all the time they are vital for the organism's survival, at least for the short time scale. In the laboratory, of course, you can maintain all of these kind of fly stocks. And this is again purely classical genetics, loss of function and gain of function genetics, to show that UBX is required to make halters. UBX is sufficient to make the halter, as well as UBX does so by modifying a wing fate into an halter fate. As you can make out, in just two experiments, they actually inferred through very fundamental concepts in developmental biology. And since the beginning of my career in 1990s, I started asking two questions. How UBX does this? UBX being a transcription factor, obviously our question is how UBX selects and regulates its targets. Because what UBX does is modifying wing fate into halter fate. So obviously many genes which are required for wing development would be modified by UBX, and their expression changes would lead to halter fate. The second question is, because of the huge difference that we see in the two organ system, it's very obvious here. One is very large flat structure, another is very small bulbous structure. But starting material is exactly the same. They are single layer epithelial cells, maybe a slightly larger group of cluster of epithelial cells for in the wing imaginary disc, and a smaller one in case of halter imaginary disc at the larval stages. But subsequently, these two develop so differently into forming two different organs. So what are the growth and patterning events that are regulated by UBX to bring in these changes. As you can make out, the one is purely molecular biology work, looking at UBX, how it binds to DNA, what kind of genes are upregulated, downregulated, how it upregulates certain genes, how downregulates certain other genes. The second is more cell and developmental biology, which uses all kinds of genetic techniques. In the first one, we used, initially we started with genetics, like enhancer trap line and EP lines and, saturated mutagenesis and so forth, looking for genes which are downstream of UBX. For this, and then subsequently with modern genomics, we started ChIP-seq, RNA-seq, and such kind of techniques to identify targets of UBX which are up and down regulated in the context of Halter development. I'll not get into those details today. In the second set of questions that we asked, growth and patterning events, here is where we started looking at, what happens when UBX is upregulated, downregulated in different parts of the developmental field, which is in this context, Halter imaginary disk. Then you ask the question that what happens if other genes are, downregulated, upregulated? So you do again classical genetics, very different mutations. You have to bring them together, use, Gal4 US system or flip FRT system to make small loss of function clones, or express, in time and space in a highly regulated way using Gal4 US system and along with Gal-AT TS system, which I'm sure all of you are familiar with these techniques. So UBX is a transcription factor of binding to minor groups. Basically has, the very simple protein, as you can make out, it's only 40 kilodalton protein. But as you can make out, such simple protein, how does it give rise to huge, difference in the developmental, context that is being to Halter transformation? So what we have identified is UBX, it simply interferes with a gene regulatory network, which are already established in the context of wing development. So very different transcription factors, bind to different parts of the genome to upregulate or downregulate genes which are required for wing development, which is evolved over several 100 million years. What UBX, during the evolution of dipterans, what UBX has done is to interfere with these ones, but not completely suppressing the appendage development by modifying it. So we have looked at very different morphogen, molecules like DPP, how DPP gradient is affected by UBX and so forth. So because functionally it is very difficult to infer which aspect of UBX function is relevant for Halter fate, because so many genes are upregulated, downregulated at the same time, because it's a transcription factor of a master gene class, right? As if the master genes means these are the genes which regulate large developmental events, not simply at a subcellular level, a transcription factor affecting one gene or two genes at a time. So what we have done is to compare UBX function across 4 gene, different insect groups: Hymenopteran, honeybee, Coleopteran beetles, Lepidopterans, particularly use the silk moth. And thanks to Shamala of Zoology Department of Mysore University, one of my PhD students actually spent some time in your department, isolating the wing imaginary disk from the silk moths, silkworm of an isogenic type. And then, of course, dipterans. Again, from all of these things, we isolated UBX and made transgenic lines expressing UBX from different insects in flies, and also ChIP-seq and RNA-seq for these developing appendages, and trying to compare UBX function across the 4 different insect groups. And we have some leads still where a lot of functional validation is going on. What aspects of UBX function, which has evolved specifically in the dipterans, has resulted in Halter evolution as against modification of a hind wing that we see in the Lepidopterans or Coleopterans or Hymenopterans. So I'll skip this. Yeah. So coming to the growth and patterning event that are regulated with UBX, again, I'll sort of look at one particular specific result that we have. So when we were looking at variety of different developmental events, one thing we started looking at is what makes wing much flatter compared to Halter. What kind of mechanobiological events. When we say mechanobiological events, basically the very different forces that are exerted on the cells, which makes cells flatter, or if the forces are not enough, they will remain, somewhat cylindrical. The Halter cells remain more cylindrical, less flattened compared to the wing cells, right? And there are scanning electron pictures of adult wing and Halter are shown here. And Halter is more bulbous compared to the two-layered, flat structure, which is wing. As you know, functionally, a flat wing is more important for flight as against a bulbous Halter, which perhaps is more important for the balancing of the flight. Now, when we started looking at what kind of developmental events which are modified in the Halter for these cells to be more cylindrical or oblong and bulbous, as against very flat epithelial structure that we see in the wing blade. And we noticed that the two major pathways, one is called Yorke pathway. I'm sure many of you know. In human, it's called YAP pathway. I'll come to human later. Another one is AKT. AKT pathway is a nutrition sensing pathway. Insulin-like signaling pathway in Drosophila, and it's also called IAS or AKT pathway. And typically, Yorke is the pathway which is downstream of multiple signaling pathway. You have heard about, in the previous speaker, talking about Wnt signaling. You may have heard about EGFR signaling, Notch signaling. Downstream of all these major signaling pathways, you have a Yorke signaling. So, and subsequent to Yorke is the cell cycle genes like cyclins and CDKs. Now, what happens is when Yorke sort of integrates all signaling pathways, some pathways upregulate cell proliferation, some of them downregulate cell proliferation. And also there are some inputs come from mechanical forces exerted on the cell surface, and particularly because of the interaction between the membrane and the extracellular matrix. And Yorke also senses those signals, and finally sends the signal to the cell cycle genes, whether the cell should undergo proliferation or not, and also at what rate it should proliferate. What should be the phase of the G1, which determines G0 and Z1, which determines how fast the cells divide and how many times they divide. So Yorke is a very important pathway across all animals, particularly in Drosophila. It was first discovered, and later it was shown to be involved in almost all kinds of cancer in human. And AKT pathway, as a nutrition sensing pathway, it actually is involved in cell size. Now, as you know, any organ shape and size is determined based on the cell number, cell size, and cell shape. Now, because wing and Halter start as a similar kind of progenitor tissue, similar kind of cell size and shape, and we started asking the question, how cell number is different? What makes the cell number to be different downstream of UBX? Of course, as you remember, everything has to be downstream of UBX because UBX is the master regulatory gene. Anything that happens in the Halter specification should be downstream of UBX. In fact, that's a fantastic genetic tools that we have. Okay, anything that we need to do, validation is important, that it has to be downstream of UBX. The second is what determines the cell size difference and the cell shape difference. And Yorke and AKT pathway, when we manipulated, right, one we upregulated, one we downregulated and one we upregulated. And we noticed that you can actually get much flatter Halter. The one which is shown here, a negative regulator of Yorke is downregulated. That means Yorke is upregulated, and AKT was directly upregulated using AKT. Both proliferation and the cell size signaling are upregulated. And we actually saw much larger Halter and flatter also. How do you know it is flatter? It's because of the way the cells are dispersed here. This is the one on the left side is the wild type wing. The one in the middle is the wild type Halter, and the one in the right is the transformed Halter. As you can see, our cells have become much flatter and sparser, or arranged very sparsely in the field, suggesting that we have actually changed the cell number as well as the cell size, but not necessarily cell shape, because that's something which we are still working on. Now, this actually, helped us to understand, at the cellular level, as you know, every time when we talk about organ size, organ positioning along the body axis, invariably, there are bigger questions, right? But finally, when it comes to mechanism, genetic mechanism or a molecular mechanism, it has to be translated into what happens inside a cell. As you know, finally a single cell is the unit of life, and all the events that happens, whether it is related to your immune system, whether related to your nutrition absorption in the gut, right? Everything has to be, done at the cellular level. So cell physiology, cell genetics, cell, morphology, cell division, everything is equally important. Even we want to, discuss the bigger picture, like, complex organism development, right? So what we are now aiming at is started with looking at, if you look at the Ed Lewis's work, for which he won the Nobel Prize in 1995, is a single gene mutation can transform the body plan of 4-winged fly or a 4-haltered fly as against a 2-winged and 2-haltered fly, right? So from there, we have actually now have tools to look at the single cell level, what are the events which regulate the halter specification. Now, coming to, what happens is, because all these genes are implicated in cancer and all of these genes are, activated at the cellular level, not necessarily at the tissue level. So we asked the question, can we use this kind of a model to address cancer-related problem? One of the problems of the cancer, I'm sure all of you know, is not just the, uncontrolled proliferation, metastasis. It's also the heterogeneity in the cancer. It's not heterogeneity between the cancer patients. It is the heterogeneity within the same individual. Within the same tumor, the cells are heterogeneous. That means each cell is independent from other, and they are behaving very differently and developing all kinds of heterogeneity. Of course, I'm sure many of you know heterogeneity is, there are 2 levels of heterogeneity. One is at the physiological level, another one is at the genomic level. So we thought, why don't we look at some of these events in human cancer and see whether we can get some insights into this, right? So if you look at YAP expression levels in many of the populations, particularly whether in TNBC, triple negative, particularly in breast cancer of different subtypes of breast cancer, increased YAP reduces the survival of the patient. That means this cancer becomes more aggressive and has poor prognosis, even if they go through all kinds of chemotherapy that is given to a particular subtype of breast cancer patients. So, and because to study this kind of genetics that we followed is so difficult in human, even if you are doing cell culture work, we thought, why didn't we use Drosophila as a model system? I'm sure many of you know that Drosophila can be used to study human cancer and other diseases like Parkinson and Alzheimer's. So, and Drosophila exhibit many hallmarks of cancer that human cancers exhibit. I'll not get into the details. So we decided that why don't we do a genome-wide screen to identify regulators of Yorkie in Drosophila, which basically leads to tumorigenesis in a particular genetic background. So they are basically the potential tumor suppressor in the human context, if the ortholog have a similar function. So what we did was very simple. You overexpress Yorkie, you get small overgrowth. In that background, you do an RNA screen. There are 12,000 Drosophila genes, and each gene you individually do an RNA screen. And then if in the context of one gene which is upregulated, which is Yorkie, another gene which is downregulated is the potential tumor suppressor. And as you know, tumor suppressors are the genes which suppresses tumor. Why do they suppress gene? Because they're negative regulators of growth. Only when they're mutant, there is uncontrolled growth. Same time, you need driver. Basically, an oncogene, it's basically a driver of the cancer. So you overexpress a driver like Yorkie. At the same time, you downregulate. We don't know which one is a potential tumor suppressor. You just basically try every one of them, right? There are 12,000 genes, you all of them. That's what it's called genome-wide screen. And what actually is interesting is, I'm sure all of you heard about, in Drosophila, what's known as giant larva phenotype. Giant larva phenotype is basically because of the increased tumorigenesis. So when the tissues grow continuously grow, the hormonal imbalance basically makes these larvae not to pupate and then continuously eat and then grow in size. And that's how you get basically a giant larval phenotype. Invariably, they die, they don't pupate, right? Or you'll have a wild type pupa. So under microscope, it's very easy to screen. You overexpress Yorkie, downregulate some X gene using RNAi. And in this combination, if you see only large larvae in the vial and no pupae, that you have a potential tumor suppressor, right? So while it is very tedious to do, at the same time, it's equally trivial to identify an interesting tumor suppressor using this kind of a screen. And if you use a GFP marker for the developing wing disc, and you can see huge, growth of these, epithelial cells covering more or less half of the larval system, right? So only Yorkie or only EGFR, and in the context of an RNAi for some potential tumor suppressor, you can see a large tumor here. Don't look at the third column, it's some other experiment. So this is how we did. We identified close to about 900 potential tumor suppressors. And these tumor suppressors, we started doing a lot of interesting bioinformatics, including some computational biology kind of work. Basically, in case, as you know, what we are downregulating is a gene in the context of Yorkie overexpression. And those genes which are causing tumor, when Yorkie is overexpressed but not EGFR is overexpressed, those genes would be context-specific tumor suppressor. If they are context-specific tumor suppressor, they must be interacting in some way, right? It's very simple, as simple as that one. If identifying genes which are related to wingless pathway or EGFR pathway, right? And if any of genes which are related to wingless pathway, they usually cluster together in terms of their functionality, right? And at the same time expression-wise too, right? So in that context, we started looking for such kind of networking of genes based on very different physiological, biochemical, and cellular functions that is already known for these genes, right? For example, if you look at these genes, and these are, by the way, these are all from the human orthologs of the potential tumor suppressor that we identified, right? And you can see that there is a clustering of, PI3-AKT pathway. Now, PI3-AKT pathway, we had already independently had shown that it interacts with Yorkie and, transforms, wing to halter towards wing. So here too, we identified a cluster of genes related to that pathway. We also identified genes, for example, right in the middle, genes related to RAS signaling, right? And that sort of suggests that, this is really the hub, for gene network related to growth control. And we also identified genes which are, related to lipid biosynthesis pathways. I think that many metabolic control genes also are implicated in growth control, which is nowadays many people are all in human cancer looking at what kind of metabolic pathways are affected, leading to cancerous growth. And lipid pathways are gaining much attention. And these are some of the genes which may help in identifying the role of lipid metabolism in cancerous growth. And they also have clinical relevance. And how much time I have? Anybody? I hope I'm audible to all of you. I'll keep talking. Professor Rangnath, how much time do I have?
15 minutes.
Okay, I'll try and finish in 5 minutes so that we can have some 10 minutes of discussion. So we did, what kind of clinical, bioinformatic analysis of the cancer genome, databases, particularly for breast cancer. So a variety of different ways you can look at it. Are these genes, particularly the human orthologs of the potential tumor substrate that are identified in Drosophila, are these associated with early cancer or early age cancer or late age cancer, or they, what kind of status they are? What's the tumor size at the time of first diagnosis? Are they metastatic or not? And how long they survive, after the chemotherapy, without a relapse, at least would they survive for 60 months or not? So, in fact, large number of them, more than 200 of the potential tumor suppressors that we identified in Drosophila and their orthologs turn out to be, very nicely correlated to the clinical parameters. Obviously, the only correlation, not causal effect. We are now experimentally validating the causality of these genes in the context of human cancer. This is a way of, you can cluster the patients depending on the expression levels. For example, the experimental way we did in Drosophila was increase Yorkie and downregulate some X gene. So similarly, you can, based on the RNA-seq data, you can identify the patients who have high levels of YAP and low levels of the gene of interest, and then you can cluster them and then see what kind of clinical parameters those exhibit, right? So, for example, a CYP4B1, which is a potential tumor suppressor in the context of Yorkie in Drosophila or YAP in human. So we found out that when this gene is less expressed in human tumors, and at the same time, YAP is overexpressed, those patients have poor prognosis. That means they have, they survived, they didn't survive very long without relapse, suggesting that this disease is, very aggressive form and not responding to the chemotherapy. So this is how we could actually identify potential new genes as diagnostic and prognostic markers, if not for any therapeutic purposes later. I'll just skip this one. So I'll just want to spend 2 minutes because this is very exciting, which also talks a lot about how classical genetics can be used. These studies are using Drosophila. Again, Drosophila genetics is so important to understand the mechanism of gene functions. So one of the set of genes that we identified in our YAP screen, our Yorkie screen, are the genes which are related to very classical promoter proximal pausing of RNA polymerase II. All of you, this is a textbook material from Bruce Alberts to molecular biology gene, whichever the book you see that when RNA polymerase starts its transcription after binding to the promoter, after a few nucleotides, 6 or 7 nucleotides, transcription happens, RNA polymerase generally pauses for some time. Unless all other cellular events are in place, the transcription will not prolong. Because an elongation step of transcription is a non-stop. It only terminates at the end, at the transcriptional termination site. So pausing is a very important step in RNA, in transcriptional regulation, right? And there's a whole complex of genes involved in pausing, right? That's what is known as, exim on LRP7 complex, SNRNP complex, we can call as. There are small RNA, 7SK RNA complex here, which is written here. And these are the negative regulators of the pausing. The positive regulator is CDK9 in response to some signal which comes from the cell. CDK9 is phosphorylated, which binds to cyclin T1. And this in turn phosphorylates RNA pol 2 and also another negative elongation factor. And this basically removes the one more steps in the pausing, and RNA polymerase II gets kinetic energy because it's phosphorylated and the elongation begins, right? So this is a very, very elegant step in pausing. And all of these genes, turn out to be potential tumor. All the negative regulator of RNA pol 2 mediated transcription turn out to be negative regulators of growth in our tumor suppressor screen. Okay, so it's very interesting. So what is the mechanism? Why is it this one, right? So we started looking at it. So is CDK9 involved in this? Because CDK9 is supposed to be this. So this is where an interesting classical genetic experiment can be done. As you can make out, what we started off with a very high-end genomics, very high-end RNA-seq, gene, such kind of genes. And finally, we did a very classical genetic screen, but using modern techniques, using transgenic lines like GAL4US system and GAL-ATTS system and so forth. Now we are back to classical genetics, right? So BIN3 is a negative regulator of the pausing. If BIN3 is downregulated and Yorkie is upregulated, we saw tumors. That's our screen. Now, if CDK9, as per this one, if CDK9 is involved in this, right, which is supposed to be the one which releases the pause by removing the negative regulators. So what happens is, CDK9 should get upregulated when BIN3 is downregulated, right? And that's how these two are the ones which relate to this tumor formation. If this hypothesis is true, what happens is you turn off CDK9 you remain hexene, let hexene remain the way there, but you upregulate CDK9 and Yorkie. So under normal wild type condition, overexpress CDK9 and Yorkie, you should still get the tumors. Indeed, that's what happens. You overexpress CDK9 and Yorkie, it can induce tumors. That means two drivers. There is no tumor suppressor here, but two large drivers. Basically, again, you can get tumors like this, right? You see the huge green blob, which is basically tumorous tissue. All others are stainings to show that they are indeed neoplastic tumors. There is, loss of epicobasal polarity, increased MMP1 signaling, and so forth. I'll not talk about those issues. But now, this is a confirmation. As you all know, in classical genetics, if you remove the function of the gene and if that particular phenotype is not there, that basically confirms the role of that particular gene. So what we did was to, we remove CDK9 in the context where hexene is downregulated or BIN3 is downregulated and Yorkie is upregulated. Now, this is the upper one is the classical screen that we did. Downregulate, overexpress Yorkie and downregulate one of the negative regulators of the growth, and you get huge tumor. In this background, you upregulate you again downregulate CDK9. If CDK9 is the mediator of this function, then you should see suppression of the phenotype. Right? It's a classical suppressor screen. And then indeed that's what we see, right? We saw suppression of the tumor suggesting that CDK9 indeed is involved in this, right? We also found out that it is Yorkie specific. So for example, if you downregulate one of the negative regulators of the pausing and you also upregulate CDK9, but you keep Yorkie under normal conditions, you don't see tumor. Suggesting that it's only in the context of Yorkie this happens. That means genes which are downstream of Yorkie is also a transcription factor. And those genes are the ones which are directly upregulated or downregulated when RNA pol2 transcription is paused. And if only that pausing when it's removed in the context of Yorkie, you see this tumorous phenotype and not general transcriptional regulation of pol2 mediated pausing, right? So this is how we actually could show that a mechanism for Yorkie-mediated tumorigenesis in Drosophila. Now we are validating this in the human cancer, whether it is indeed human also. And again, the classical gene, the breast cancer or any other cancer genome analysis clearly suggests that even the genes which are involved in pausing, when they are downregulated, they show poor prognosis on the patients, suggesting that there may be potential tumor suppressor even in the human cancer context. And now we are validating them using the clinical samples in collaboration with clinicians. I'll stop here. And these are the students and postdocs who worked over several years and who contributed to all of this work. And, fantastic team. As you can make out, the amount of work that goes in all kind of these requires almost like a human chain. Many people have worked on this other than the people who are on this picture. Thank you very much, and thanks for the opportunity to talk to all of you. I'm happy to take some questions if there is some time. Thank you.
Obviously, thank you for an illustrated by you very nicely, starting from UBX and audio development and how exactly the basic understanding can be extrapolated to the understanding of the cancer and whether to negative regulation or positive regulation. Thank you on behalf of the Department of Genetics and Genomics and on behalf of the university and on my own behalf. Thank you very much. And we have some questions from our students and can ask, you can. I have one question.
Yes.
How Yorkie, if you downstream of Yorkie, if you just knock out this Yorkie, what about the downstream of genes which are there, which is involved, or which is depending on the Yorkie regulation or Yorkie?
So what? Yeah, thanks. So you see, in the genetic screen, there are two ways of looking at it. If we do RNA-seq, when you upregulate Yorkie and see what are the genes which are upregulated or downregulated, like you have two tissues. Normal tissue and the tissue which has Yorkie is upregulated. And you do RNA-seq, we did this, and we identify genes which are differentially expressed. Some genes get upregulated, some get downregulated. but which one are they functionally relevant to this? So this RNAi screen that we did, the classical genetic screen, helped us to identify functionally relevant genes which are associated with the Yorkie function. But not all of them would be downstream of Yorkie. Someone can be in parallel to Yorkie, but definitely associated with Yorkie because they are context specific. So now what we have done is we compared the genes which are part of the genetic screen, also as part of the RNA-seq information. Those are the genes which are the potential downstream of genes which are mediating the Yorkie tumorigenesis function. So we are validating them at the experimental level.
Any other question from our students? In India, we want.
Shashi, thank you very much.
Thank you very much. Yeah.
Talk for the students. I'm really honored from your presentation. Thank you, Shashi.
Thank you. Thank you all.
Okay. Thank you, Professor Shashidhar, for your time. I know your schedule, very busy, with all busy schedule, you are here to enlighten our students. Thank you very much.
Thank you. Thanks.
Shakuntala,
Sir, please. Heli, sir.
when Shashi asked how much time I have, because nobody answered, I told him 5 minutes.
ಸರ್ ಅದು ನಮಗೆ ಇದ್ರಲ್ಲೇ ಬಂದು ಡಯಾಸ್ಗೆ ಬಂದು ಮಾತಾಡ್ಬೇಕಿತ್ತು ಸರ್. ಇಟ್ ಟುಕ್ ಸಮ್ ಟೈಮ್ ಫಾರ್ ಮೀ ಟು ಕಮ್ ಟು ದ ಡಯಾಸ್.
On behalf of all of you, I said 5 minutes. I don't know whether it's okay.
Yeah, it's good that you mentioned 5 minutes so that I could sort of speed up. Otherwise, I would have gone for another 10-15 minutes.
Anyway, Shashi, excellent talk. As a good teacher.
Thanks, Professor. All your training.
Oh, God.
Inspiration.
Okay, thank you, sir.
Hey, Meva is here. I didn't see him. You're on mute, Meva.
Now I'm allowed.
Yes.
Otherwise, I was forced to mute.
Yeah, of course.
Thanks, Shashi.
Good to see you.
I was there in the hall, but I had to sign some papers, so I came here and put it on through Zoom.
See. Okay. Rangnath, are you in Mysore or in Bangalore?
Bangalore, sir. Everything online now. Because I have some contingency at this end, I couldn't leave it.
ಏನು this age? ಏನು age ಆಯ್ತು ನಿಮ್ದು? This age ಅಂದ್ರೆ ಏನು?
Shashi, the book is almost ready now, e-book.
ಮಳೆ ಮಳೆ ಇರ್ಬೇಕು
Okay, I will hear from you. You understood my question, sir? My question was very simple. You look at this central dogma, you have DNA, RNA, and so on. Right? So now, what I'm trying to say is that RNA is the RNA. Not just in chemical contact, you have a sequence of what will be the content of the cell and what will be the control, right? Using that to the cell. No drivers, no drivers. So the people that will be prepared, what will be there? No, getting. Any answer?
Excuse me, sir. Yeah, this side. Sir, maybe the RNA, mRNA which comes out of the cell, it may get damaged when it is coming out of the nuclear membrane. No, it is the advantage I'm speaking about.
Yeah,
I'm speaking about the advantage of mRNA coming out of here.
mRNA is not possible. You have to directly translate the message from the DNA. So, what is the advantage?
Okay. So due to network problem in Trivandrum, so Dr. Monek Banerjee is unable to log in right now. We'll wait for another 5 minutes for him to join. Thank you. Thank you.
Now, which name you are allowed?
Hello.
Hello.
Can you hear me?
Yes, sir. Yes, sir. We can hear you.
Okay.
Good
Yes.
afternoon, Dr. Monik Banerjee.
Good afternoon.
Good afternoon. Yeah. I'll introduce you to the audience present here. Dr. Monik Banerjee is a senior scientist at the Rajiv Gandhi Center for Biotechnology, Kerala. He obtained his PhD from ML Sukhodia University, Udaipur, Rajasthan. He is a human geneticist working mainly on the intricacies of neurophysiatric disorders, including diagnosis and therapeutics of these diseases from genetic, pharmacogenetic, immunogenetic, and epigenetic perspectives, and is incumbent president of the Indian Society of Human Genetics and member of American Society of Human Genetics and International Genetic Epidemiology Society. Recipient of BOL Travel Fellowship and International Fellowship of Brain Science Institute, Tokyo, Japan. And apart from this, has a very good academic records. And I welcome him wholeheartedly for this session. And thank you very much, sir. Over to you, Professor Monik.
Thank you very much.
You can present
I
your.
was
Yeah.
Welcome.
Are you able to see my slides?
Yeah.
Yes, sir.
You can see
Yes,
you
Yeah.
and
Yes, sir.
Right. Thank you very much. It was indeed a pleasure receiving this invitation. I was connected since morning. All of a sudden, when my talk came, it got disconnected there. So anyway, so without any delay, I will straight away start with the work, the topic that I have chosen to speak today. We had been working on genetics and epigenetics of various complex disorders. So today I will be restricting my talk only towards the how much do we really understand about the epigenetics of schizophrenia, and with whatever we understand, how much is the real significance with respect to its pathogenesis there. So that is where I start with. We all know that yes, all complex disease, whether it is schizophrenia or any complex disease like diabetes, there is a huge component of environment. Now, restricting that particular environment to just seasonal changes, you can see that the children born during the month of the winter months are more prone to develop autism, schizophrenia, coronary heart disease, and various other cardiac issues there. So one would wonder that yes, seasonal changes do influence the pathology there. And seasonal changes, in addition to his genetic component, in addition to other environmental factors, which allows the particular gene to express whether aberrantly or normally, that will decide whether we should be going towards a course of event that which may not be favorable, whether be it for schizophrenia or mental disorders, or be it for diabetes, heart disease, or anything of that sort. So all these complex disorders actually have a very strong component of the genes as well as very strong component of the environment. And this is how interaction of these tries to influence the gene expression patterns. Now, how do we understand the gene expression pattern? It is not about the gene expression alone, but gene expression is actually mediated by several other factors, which we call as epigenetic factors. And so therefore, to understand the gene expression pattern, we really need to understand the histone modification aspects of the cell, the microRNAs of the cell, the DNA methylations of the cells, and then see whether these factors, to what extent do they influence the gene expression pattern there. And these are the factors which are extensively being studied with respect to pathogenesis of various complex disorders, including schizophrenia there. But what we started, so when we decided to work to take up this issue of epigenetics in schizophrenia, what was already known was, yes, it influences the global methylation patterns, but there was no consistency between the observations. Someone has reported hypomethylation, someone has reported there is no correlation. Then there has been reports of hypermethylation status of various genes. Then there has been also reports of hypomethylation of some genes. Then of course there are newer methods of studying genome-wide methylation aspects. So all these things literally reflect that yes, there are epigenetic changes do influence the pathogenesis there. But what is the main concern in all the studies is they had all been taken patients from a conventional treatment background there. So it means that the patients were actually undergoing medications there. So no one has really worried about the influence of medication aspects in understanding the epigenetic patterns there. So we questioned all the studies that has been coming up for the last 30 years, that are we literally clear about the epigenetic signatures that has been reported so far? Are they the real signatures of pathogenesis, or they are the drug-induced signatures of schizophrenia there, or treatment response there? So questioning all these historical papers is really not an easy job. So we literally, once we question it, we also have to prove that whatever we are questioning is really on the right direction there. So this particular perspective or criticism of all the studies was published in a specialized journal of epigenomics. These are various other studies. And what people have done mostly is by taking the study, they have mostly compared with the cases and the controls there. So they have taken the control samples who possibly don't have schizophrenia and those who actually have schizophrenia there. So when I look from that point of view, I can see that my patient actually there is a hypermethylation status. And when I see with respect to homocysteine, which is another marker for methylation, I see that there is a hyperhomocysteinemia in my patients. So one would wonder that if I literally stratify my patients based on these cases in the control, am I really doing justice towards dissecting the pathogenesis signature, or am I literally being worried about the influence of drug-induced alterations? So, but in a clinical scenario, it is not possible because the patients will be undergoing various types of medications. So therefore, they will not be under a single medication there. It is just next to impossible that I can really say that this particular patient is actually taking on this particular medication there. And even getting patients from the tertiary care hospital is also questionable because we don't know whether this tertiary care hospitals, at what level did this patient reach those tertiary care hospitals there. Because most often the psychiatric illnesses are actually treated randomly at home by the known doctors. And once they are beyond their control, only then they reach the tertiary care centers there. So therefore, with these issues, we thought that we will try to investigate whether these medication issues do they really influence the host epigenome or not. So before I get into that, so we always think that whatever medication that we are taking is really safe there. But if you literally look into these slides, so this is the paper that we published, where we actually questioned all those papers that have been published till date for the last 30 years. And it was about the crossroads of epigenetics and pharmacoepigenetics. And with these hypotheses, we actually try to investigate whether these medications are actually influencing the DNA methylation status. Do they influence the histone modifications? Do they also influence the microRNA expression pattern or not? So generally, it is believed that medication is very safe, but let us not be in a fool's paradise, that medications are really not that safe, because on an average, the 50% of medications that we routinely take are actually likely to be ineffective or are non-responsive to most of the patients. And at the same time, you can see that there is a strong association between antipsychotic polypharmacy and high dose prescribing medications. So medications, and similarly, what is the medication that we are really looking for? Like these are the forms of medication and the side effects that were reported before 2000, and this is beyond 2000 there. So are we literally taking the medications pertaining to these side effects, or are we taking the medications that actually reflect to these side effects? If you literally go to the typical government hospitals, I will presume that the patients from the government hospital will have these type of side effects, whereas the patients from the tertiary care or the private hospitals will have this type of side effects. Because we have shifted from the typical antipsychotics to the atypical antipsychotics now. And this is the side effects of the atypical antipsychotics. Am I audible, right?
Yes, sir.
Right. Then another issue is that we really don't worry about even the paracetamol. In fact, this was one of the largest study that has been carried out, which says that even paracetamol and Prof.N.B, which we normally take, or aspirin, that can also result in serious pregnancy-related issues, and it can also result in the risk of cerebral palsy in the child there. So the routine medications that we take also can induce a lot of side effects. Or similarly, these are the issues where most often we also are not aware of it. We have been working on this particular aspect, where anti-epileptic drugs, which are routinely taken by the pregnant mothers, and these anti-epileptic drugs can induce various types of malformations even in the offsprings there. Many of these things are believed to be of congenital, are believed to be genetic in nature, like cleft lip, various heart valve defects. And ideally, one should really, before taking the cleft lip patients, before taking the heart valve defect patients, one should really worry about, are these defects being induced because of the medication that the mother was taking when she was pregnant there? So these medications that we routinely take, whether it is anti-epileptic drugs, antipsychotic drugs, or psychiatric drugs, or whether it is routine paracetamols, what has not been tested so far is teratogenic potential there. Even the FDA approved drugs, 91% of the FDA approved drugs have not been tested for its teratogenic potential there. So that really raises concern that, are we literally safe enough there? And one would really say that, why should I literally worry, because schizophrenia happens at later part of my life. So therefore, there is no point in really testing antipsychotics or antidepressants for its teratogenic potential there. But I'm sure that who are working in schizophrenia or depressions and all, even we have patients at the age group of 16 to 17 years there. So therefore, whatever medications, whether it's antipsychotics, antidepressants, or paracetamol, all the drugs should be tested for teratogenic potential, and which has not been tested even in the FDA approved drugs. So this literally raises concerns that, are we really safe enough for whatever medication that we take in there? So with these, we decided that we will actually first try to work it out about the effect of individual antipsychotic drugs under the in vitro conditions, and then validate those observations under the in the patient group there. And the patient groups we have divided into responsive and non-responsive there. So we have taken both liver cell lines and also neuronal cell lines, and then we have treated with various antipsychotic drugs, clozapine, haloperidol, and olanzapine. These are mostly routinely given drugs there. And then we checked for global methylation analysis. We have looked for histomodifications. We have looked for microRNA. We have also looked for epigenome-wide methylation and aspects. And then we also looked into the gene-specific expression with respect to epigenetic genes and the pharmacogenetic genes. So whether these methylation patterns, histomodification of microRNA, are they literally reflecting alterations with epigenetic gene expression patterns or pharmacogenetic genes, and then validate these observations in a subset of the patient groups. So these were the set of genes. These are the global methylation analysis that we have done. We have mostly looked into the 5-methylcytosine, 5-hydroxymethylcytosine, 5-malcytosine, and then so which are the indicators for methylation as well as your demethylation status. And then we have also taken the genes that are responsible for methylation as well as for the demethylation status, like DNMTs and TETs and all that. And then we look into the protein expression of these genes. And then we also considered, so that was all about epigenetic genes. And then for the pharmacogenetic genes, we have taken various cytochrome P450 genes, ABCB1 drug transporters. And then we have looked into histone modifications. Then we have looked into the CPG site, methylation sites for all these genes. And then looked into the microRNA expression profiles, and then target validation of those microRNAs for the specific genes. And so this goes through a series of observations, and if anyone is interested to go into the details of all these observations, we have already published this paper. This one you can actually verify it in the manuscripts, published in the Epigenomics. So where we observed that there is an hypermethylation, there is a deoxymethylation also. Then the haloperidol, clozapine, and olanzapine actually results in increased DNMT expression, DNMT3A, MBD2, TET1, and TET2, which to somehow explains that overexpression of these genes possibly might reflect in increased methylation or hydroxymethylation statuses of the cells. And this was further verified with respect to combination of drugs. And then we try to look into concentrate only with respect to DNMT. So, and then there's one particular microRNA which specifically binds to DNMT1. We found that the microRNA 29B actually is downregulated, which literally explains that upregulation of these expression might be possibly mediated because of the downregulation of microRNA because of the medication aspect, because of the haloperidol, clozapine, or olanzapine. So therefore, these medications actually induce downregulation of microRNA, and which in turn results in upregulation of various epigenetic genes and overall global methylation patterns. So this is clearly evident here also, which is a summary of all this observation. That global methylation there is an hypermethylation status, there is an increased gene expression pattern, and there is a decreased microRNA MYT29B expression patterns. Then we try to look into the same observations with respect to liver as well as with respect to neuronal cells. Almost, so this is all published in Epigenomics in 2018 and in PLOS One 2017, where whatever we have observed in the liver cells, they are also being replicated in the neuronal cells. And then we wanted to see whether how much of these observations that we get from the in vitro data can actually correlate with respect to the patient scenarios. So what we did is we collected the patient's PBMCs, and then we try to stratify. We try to get the patients who are actually classified as to responders and non-responders, and then checked whether what is the PBMC, what is the methylation status, and we have also selected these patients based on the DNMT1 mutation background and the ABCB1 mutation background. So the DNMT1 mutation background, the reason why we have chosen is because this particular variant has been reported to be associated with with schizophrenia itself. So therefore, we have chosen the patients in such a way that they have this particular DNMT1 genetic background also. And what we see is that there is an hypermethylation status is actually more reflected with respect to the drug response there. With non-responders, we really don't see there is much of a change with respect to the controls, but hypermethylation status is more associated with the drug response there. So therefore, what we when we stratify the patient group, when we just try to do the study between the cases and the control, what we see is the hypermethylation status in the is associated with the patient group there. But here, the hypermethylation status actually reflects with respect to the treatment response there. So then we wanted to see whether this is with respect to epigenetic genes. This is what has been shown earlier, that the hypermethylation status with the global methylation status. Similarly, we see the increased DNMT expression in the patients. In the responder groups, increased DNMT3 expression, increased DNMT3B expression in the patient group, in the responder groups, and there is a downregulation of MIR29B. However, there is a further downregulation of MIR29B in the non-responders, which we are yet to understand why there is a further downregulation of MIR29B expression in the non-responder groups. And here we have not controlled for the medications there. The patients are actually selected based on the naturalistic background there. It means that the patient had been taking medications, what the doctors have been prescribing there. And most of them had been taking, had been given multi-drug therapies there. So anyway, with respect to drug response and our in vitro observation actually very well correlates there. So medication possibly might be acting through the epigenetic routes. Then we wanted to investigate whether these drugs also influence the pharmacoepigenetic genes. Pharmacogenetic genes. Pharmacogenetic genes are those which are responsible for metabolizing the drugs, like the drug transporters or drug metabolizing enzymes on the drug target genes. So here also what we see is that with respect to haloperidol, clozapine, and olanzapine, they upregulate the ABCB1 expression, they upregulate the CYP1A2 expression, they upregulate the CYP3A4 expression, and haloperidol also upregulates CYP2D6 expression, and even the combination treatment also increases the expression of all these genes. And then these haloperidol, clozapine, and olanzapine also downregulates MIR27A and MIR128A. Now, what is the role of MIR27A and 128A? MIR27A actually regulates the expression of ABCB1 and CYP3A4. So, upregulation of these genes might be mediated because of the downregulation of these microRNAs with respect to medications, and upregulation of CYP1A2 overexpression is also associated with downregulation of MIR128A expression. So this is what we observe, that these antipsychotic medications, whether it's clozapine, haloperidol, or olanzapine, results in downregulation of microRNA expression. And this we try to validate further in the clinical scenario. And in the clinical scenario, what we get is, yes, ABCB1 is overexpressed, and this overexpression one can, with respect to responders, but with the non-responders, you can see that in the microRNA, MIR27A is actually much more downregulated there. So ABCB1 overexpression can be possibly related to the MIR27A expression, but CYP1A2 expression, which is downregulated in the patients, we actually don't see any significance with respect to the MIR128A expression in the patients. So, however, it associates with the drug response. We are yet to understand why these non-responder groups behave differently. So, but yes, one can really say that medication or antipsychotic drugs are actually mediating drug response through the microRNA directed activity. So then we wanted to see, so these are about individual microRNAs. Then we wanted to see whether what happens when we try to look into the entire microRNA expression. So then we have taken close to 900 microRNAs. But before going into microRNA expression, what we published is that these were the microRNAs that has been reported to be altered in schizophrenia. So these are the microRNA which were reported downregulated. These were the microRNA that were reported upregulated. But then we did an in silico observation that how many of these microRNAs are actually being regulated by antipsychotic drugs. This was also published where we found that yes, there are antipsychotic drugs which regulate large number of microRNAs. That was through in silico observation. So this is reported with respect to pathogenesis, and what we questioned this expression of microRNA with respect to pathogenesis, and we say that yes, these microRNAs might be regulated because of the antipsychotic drugs. And then we try to check whether those microRNAs are really being regulated because of the medication issues. And we found that yes, there are a large number of microRNAs which are upregulated, which falls into various categories like epigenetic machineries, ABCB transporters, drug metabolizing enzymes, and of course the various metabolic genes. So there are so many microRNAs which are upregulated, which belongs to these categories, and there are so many microRNAs that are downregulated in with respect to antipsychotic drugs. So and then we try to this is almost the same, but showing in a different representation there, and then the overlap between the observations between the various antipsychotic drugs and the microRNAs. And what interesting is that now we observed is Mi29B was reported to be downregulated earlier, and now we say that the Mi29B is also associated with drug response there. So all these microRNAs has been reported to be altered, and these were the microRNAs which has been reported to be associated with schizophrenia. And now what we say, all the yellow ones which you see is our observation. So where we say that yes, these microRNAs that have been reported to be associated with altered expression in pathogenesis. Now we say that those microRNAs also have a role in drug response there. So that literally questions all the microRNA observation that has been reported till date, because which really says that those microRNA expression might be influenced because of the antipsychotic drugs. So with this, we talked about microRNA expression and the various epigenetic genes and pharmacogenetic genes which are influenced because of medication. Now I'll try to change gear slightly to see whether these medications do this schizophrenia also are influenced because of these epigenetic genes. So do this genetics of epigenome, do they really directly relate with respect to schizophrenia pathogenesis? To address this, one has to literally understand, do these methylation changes, do they have ethnic basis or not? So this you can see that yes, they do have an ethnic basis, where the blacks has been reported to be extremely, there is a very high methylation status in compared to the Hispanics and whites then. So this is when this has been published long back by some other groups. And what has been reported with respect to gene-specific or chromosome-specific methylations, this is for the schizophrenia set. This is with the normal controls there. So you can see that there are different sites of methylations with respect to schizophrenia and the normal controls. This has also been published by some other group. We said that how much of these methylation changes are really being influenced because of and can this methylation changes be influenced because of the ethnicity or not? So let us take that some of those methylation changes that has been reported. So here, these are the methylation CpG sites which are reported to be altered. And what we see is that this is the hypermethylation state that you try to see. This we extracted the 450K methylation data from the public domain, and we try to see whether do they literally reflect in the ethnicity in a similar way or not. Let us consider on this particular methylation site. So this particular methylation site is reported to be hypermethylated in the Caucasian groups, and what we observe is that in the African groups, this particular methylation site is like to be hypermethylated there. So these are the regions where you see that the hypermethylation in whites actually is in contrast to the Africans' hypermethylation states there. So this possibly might reflect a gene-specific expression pattern with respect to ethnicities also there. And does it have any specific genetic basis also or not? So to understand the genetic basis, we try to understand the genetic variants for the methionine cycle genes, which are the exogenous control of methylation, and then the endogenous controls where the DNMTs and the demethylations like TETs, MPDs and all that. So, and then we try to see that yes, DNMT variants are directly associated with schizophrenia. So some of the variants which are directly associated with schizophrenia. And what is the role of these variants? These variants are actually associated with increased methylation status there. So these are the two variants which are actually reported to be associated with schizophrenia, and both of them actually relate with respect to increased methylation status. And that might possibly explain the altered methylation status between the case and the control there. Similarly, with respect to exogenous control of methylation, where the folate pathway gene comes into play, here also we find a couple of folate pathway genes that are actually associated with schizophrenia directly there. And here also we say that yes, variants in the folate pathway genes may contribute to increased serine levels and decreased folate levels in the schizophrenia subjects. And I'll show you why this is very important to assess, because do these antipsychotic drugs also deplete the folate levels or not? So if these antipsychotic drugs deplete the folate levels, then the folic acid supplementation will have to be based on his genetic background also. Many of these drugs, like particularly anti-epileptic drugs, are known to deplete folate levels. So therefore, for pregnant mothers who are on anti-epileptic drugs, they are actually supplemented with folic acid there. So say similar scenario would be applicable here too. So implication of these this folate pathway genes association might possibly explain why there is an hyperhomocystinemia in my patient group there. So, which means that the genetics of epigenome is also very critical in understanding the role of these genes with respect to methylation status, or with respect to demethylation status, or with respect to medication issues. And this will also determine the threshold of environmental factors that actually will play a role in influencing these factors, these environmental factors. And do antipsychotics enter? So then we came across this particular set of papers where we observe that yes, antipsychotic drugs do interfere with one-carbon metabolism, and there are reports that antipsychotic drugs actually can influence the one-carbon metabolism, and even which can also result in vascular dementias also. And some of the metabolic events that are associated with many of the antipsychotic drugs can possibly be also explained because of these epigenetic genes or the alteration of expression of epigenetic genes there. So therefore, antipsychotic drugs which are believed to target mostly the neurotransmitters, one would really say that whether the response that you're getting, are you literally getting the response because of the right targets of the neurotransmitters, or is it because of some other mechanisms there? On one aspect, we prove that yes, it could be because of some other mechanisms, which could be through the epigenetic events, modulation, or does it also influence the therapeutic response through immune suppression there? So in another article where we published in Journal of Neuroinflammation that a couple of cytokines, mostly the pro-inflammatory cytokines, are genetically associated with schizophrenia there. So therefore, and these association of this pro-inflammatory cytokine will possibly result in increased quinolinic acid, and increased quinolinic acid will result in increased NMDA-induced toxicity, or it will also result in increased glutamate level, which will further increase the NMDA activity there. And therefore, the antipsychotic drugs possibly might also play a role through the immune suppression. And in fact, well, this I came through when I was discussing with my clinician. So he said that he was surprised to know that when he was giving his patients just anti-inflammatory drugs, some of the patients actually were responding there. So that was we were very curious, and then we found that yes, antipsychotic drugs are particularly haloperidol and clozapine has been reported to have highly immunosuppressive events there. And people in the West, they have also tried that this particular immune suppressive drug has been given in schizophrenic patients too. So with this, I come to an end if I have not exceeded my time. So what I said is completely a different understanding so far that we had it with respect to epigenetic events. And this is applicable not only with respect to schizophrenia, this is applicable for all complex diseases, because all complex diseases, the patient come for research aspects or come for understanding the biochemical parameters when they are undergoing in the conventional medication treatment. So what William Osler said, that made an observation that if it was not for the great variability among individuals, medicine might as well be a science and not an art. And today we are trying to understand this art of medicine through genetics, epigenetics, and pharmacoepigenetic events. So a large part of this work was actually carried out by Swati, and this is Sharada Lakshmi, so who contributed towards the understanding of pharmacoepigenetic events and the epigenetic genes in schizophrenia. With this, I come to an end. I would be very happy to address any queries if you have. Hello.
Dr. Manekman, our illustrious lecture given by you on epigenetic modification of antipsychotic drugs and its teratogenic effect. It was really a wonderful lecture, and really it's upcoming one, right? Recent one, epigenetics, because it's near a decade. We started with the epigenetic one, and now it is extending everywhere in genetics and genomics and so on. It is very interesting. And I have one question for you.
Sure.
Is this epigenetic modification that you have taught, the demethylation and all, is it temporary, or when there's a psychiatric drug withdrawn, can it be reversible?
Yes, it is reversible there. But to what extent it is reversible, it is still not understood, because now it is believed that epigenetic events are also inherited there. Okay. So therefore, it has been reported that mothers who are actually undergoing stressful childbirth, it will undergo a lot of epigenetic events because of the stress factors, and the offsprings of these stressful mothers can actually result in altered epigenetic events in the offsprings, and which can result in development of schizophrenia at a late onset, or even an autism at the early onset.
Okay. So, is it something with continuous antidepressant?
Yes. No, pardon, I could not hear you. Can you repeat?
Is it antidepressant also have the same effect, epigenetic effect?
Is it? Can you repeat again?
Antidepressants also have the same epigenetic effects?
Yes. Actually, most of these drugs now, we were the first one possibly in the world to talk about the pharmacoepigenetic of valid. People had been talking about pharmacoepigenetic events, but no one actually has tried to explain it with a valid experimental design there. So we had been working extensively on this pharmacoepigenetic events, and now we have a very clear observations with respect to pharmacoepigenetic events. And yes, many of the drugs, whether it is antidepressants, antihypertensives, they all actually have pharmacoepigenetic events.
Thank you.
Right.
Onat?
Yes, sir. Yeah, tell me. Yeah.
Thank you for agreeing to give talk, and with all the incident and the issues, challenges, we could able to give a good talk. Thank you for joining for this event, and I'm highly impressed. Thank you.
Thank you.
So on behalf of Department of Genetics and Genomics and behalf of the University of Mysore and on own behalf, I heartily congratulate you for a nice lecture which is given by you and really useful to our students. Thank you very much, sir.
Thank you very much. Thank you very much. Thank you.
Well, that marks the end of the third session of this conference. And with this, I would like to thank the session chairs, Dr. Amritavalli and Dr. Shakuntala. I request you to come on the dais and receive this bouquet.
Friends, after the lunch, you wanted to come back and sleep here or sit here because they might talk. They told me to give at 2 o'clock. Now it's already 1:30. I don't think if I rush too much for you to have lunch, any problem? Can I give talk or not? So in that case, I think organizer will agree. 2:10 will start. Okay? By 3 o'clock, I complete my talk. If they give time, I can talk 6 hours. My long standing for Gulbarga University student 6 hours. Okay, fine. Then total they'll come back 3 o'clock.
Okay, so I request Ashita to honor the session chairs. Huge round of applause, please. Thank you, everyone. Also, another announcement. So for the certificates, students from MSc Zoology, if anyone has not registered their name yesterday, you can meet me. I'll be here for the next 5 minutes. And anybody other than MSc students who are here,